Hello, everyone. Good morning, good afternoon, good evening. Thank you all for joining in at all these different, from all these different time zones. My name is Ilana Krista. I'm a professor in the Department of Molecular Biology at Princeton University. And I have the privilege of being the chair of the Biology and Disease-Driven Human Proteome Project. And it gives me great pleasure to welcome you to this inaugural webinar that we want to be part of a series of webinars that we are working on. And this webinar is dedicated to post-translational modifications and understanding their role in human disease. I'm just going to start by thanking my co-organizers, the other members of the executive committee of the Biology and Disease Human Proteome Project. They have put a lot of effort very quickly in making this webinar possible. And I also want to thank our tremendous help for administration and promotion of this webinar from Mary and Alan from the Princeton University Administration, Chelsea and Amanda, who helped us to advertise this webinar, and Vera, who is the chair of HipA Marketing and Membership Committee, who put together the flyer that you all saw. So this, as I said, we want to is going to be just one of a series of webinars that we are planning as part of the broader Human Proteome Project. And so these future webinars that I encourage you to keep an eye for will be organized by the pillars of HPP, which means the mass spectrometry, antibodies, and knowledge-based pillars, as well as by the C-HPP, which is the chromosome-based Human Proteome Project, as well as by us, the D-HPP, the Biology and Disease-Driven Human Proteome Project. And I want to encourage you to get involved in these webinars. We want to have a series of webinars that is truly interactive and that fits with the research directions of this proteomic scientific community. So several webinars are being planned. I can tell you already that one theme that we are trying to put together is the proteogenomic webinar focused on this area of research. But please contact us in proposed themes. We want to hear your thoughts, also your thoughts about the format of these webinars. We want to make them as interactive as possible. And we also want to encourage attendance of scientists from different career stages. So even the speakers for today, as you'll see, also are speakers that are at earlier careers, junior or early career investigators and senior career investigators. Also, we want to make this as interactive as possible. And that's why at the end of the talks, we have this panel discussion. Contact us and let us know what other formats we should consider. We are all ears. We want to hear your thoughts. We want to involve all the D-HPP teams that focus on different areas of human health and disease, the broader HPP community, and the broader proteomic scientific community. So for today, this inaugural webinar is dedicated to post-translational modification. And this was a theme that was really straightforward for us to select. We all know that post-translational modifications are these important regulatory hubs that can control protein structure, protein interactions, subcellular localization of protein, and really critical for the making a protein accomplish so many different functions, allowing a protein to do all this. We all start to understand that PTMs are diverse and dynamic in nature, and that this breadth and depth of PTMs can be quite daunting. It seems like every year we learn not only about the new type of post-translational modification, but also how proteins can be decorated by so many different modifications. And we start to have the technology in place to detect all these modifications, but we still know quite little about the function of all these modifications, or how they interact, how they crosstalk, how they coordinate the protein function. But we start to understand how important these PTMs are in health and disease. So with this in mind, we invited a series of speakers who focus on different types of post-translation modifications or on developing the bioinformatic tools and platforms necessary for discovering PTMs, even novel PTMs. And we thank our speakers for accepting our invitations. We are really delighted to have such a strong panel of speakers. And with this, here is the program. I'm gonna pass this to Ferdinando, who is gonna tell you a little bit more, provide you a brief perspective on proteomics and PTM studies. So Ferdinando, you can share your screen.
Yes.
And while Ferdinando is doing this, after Ferdinando, we'll start with the speakers, and our first speaker will be Nicki. And each talk will be 15 minutes, and then we will have 5 minutes dedicated to question and answers. So I want to encourage you all to please type your questions in the Q&A below, and the chair for each speaker will read those questions to the speakers. So Ferdinando.
Yeah, thank you, Now. So while we were preparing these webinars, an article appeared in the Annals of Oncology. It's a recent publication from some days ago from a group from UCSF and University of Oregon. Annals of Oncology is a flagship journal of the European Society of Medical Oncology. And this is actually an update estimate of cancer patients who could be eligible and can respond to genome-targeted oncology drugs in the US. There were previous studies from the same group published also in other clinical journals like JAMA, for example. And we found this could be somehow interesting because if we look at the two main figures of this manuscript, we see that between 2006 and 2020, the number of patients who were selected to be eligible for genomically targeted therapy increased from 5% to something more than 10%. The different colors are the different cancer which has been studied. And if we look at the number of patients who responded to a genomically targeted therapy between 2006 and 2020, we also see an increase between something around 2 to 3% to something which is around 7%. And this is actually the numbers which are reported in the study in the abstract. So if we look at the time period of 14 years, 2006 and 2020, the number of patients who were eligible for a genome-targeted therapy increased from 5 to 13%, meaning that actually 85% of our cancer patients are not eligible for genome-targeted therapies. And the response to these targeted therapies based on genome were increasing in this 14 years between 2.7% to 7%, meaning that 90% of the patients do not respond to genome-targeted therapies so far. I dig a little bit in the supplementary of the paper and look on the roughly 50 drugs which were approved by the FDA for genomic indication. The majority of them are TKI, and we expected them. We had some antibody-based drugs and some other small molecules which were not directly targeting post-translational modifications. This is maybe stating the obvious, but together the information we have seen in the number of eligible patients and responders and the kind of drugs we have seen from the graph before seems to highlight a gap between the biological information we are addressing to take decision on the treatment to give to our patients and the biology we are targeting with our treatments. And probably filling up the gap is something that could help to advance and to realize the promise and the potential of precision medicine. And it's very likely something that our community and our expertise can contribute. And that we've thought that could be an interesting aspect to discuss in the discussion part of the webinar. That said, I'd like to introduce the first speaker of the webinar, which is Professor Nicki Packer. Nicki is very well known in our proteomics community. She's professor at the Department of Molecular Science in Sydney at the Macquarie University, Australia. She has spent a lot of work and efforts to build up strategy on glycomics, glycoproteomics. He definitely is a big reference in that field. And she also putting focus on how this information can be interpreted in the context of disease and the biology who could be applicable for improving the way we treat disease. So I'm really very much looking forward to your talk, Nicki, and I'll leave the sharing to give space to your presentation. Oh my God. There we go.
Okay. First of all, I'm absolutely delighted to be doing this because I've been involved, as most of you may know, for a very long time in proteomics and glycomics. And for it to be post-translational modifications coming into focus in the HPP it's very exciting. So I'll just move on, I hope. Yes. This is my favorite slide, which shows why we are interested in glycomics or sugars. So this is a beautiful picture of the hairiness on the surface of a cell, endothelial cell in this case, and the glycocalyx, which is the first thing which any other cell that passes by sees, and it's nearly all sugar. Obviously, it's mostly as glycoconjugates, including glycoproteins, and over 50% is estimated to be glycosylated of the human proteome. So all I can say is it's high time that glyco came into the proteomics world. This is something that you've probably seen if you've looked at the textbook Essentials for Glycobiology. And this webinar and this group focuses on glycoproteins. But I just wanted to make it clear that there are other glycoconjugates that we should start looking at as well. Now, the difference between glycoproteins and proteoglycans is really quite large, even though the words mean the same thing. But they still are presenting these sugars to the surface of the cell. They're looking at the outside of the cell and they're interacting with everything around them. And of course, there are these other little things with single monosaccharides on them, which are inside the cell, but we're not even going to talk about that at this stage. So this is something which we calculated a very long time ago. What difference does glycosylation make to the proteome? If we estimate there's about 50% N or O glycosylated, that gives us, and I'm not going to do exactly, but about 10,000 glycoprotein genes. If we say 3 sites of NO per glycoprotein, we get 30,000 sites of glycosylation. One or all of these sites may be occupied, which gives you 8 combinations, which gives you 240,000 glycoforms of proteins. If at each site there may be up to 3 structures at each site, we get more than 6,480,000 possible glycoproteoforms of the genome of the cell. And that's, I think, a vast underestimate. So this might be why the genome doesn't account for all the therapies that we try. So as Ileana said, or might have been Frank that said, I can't remember who said it, but the technologies are there now. They're not all there, but they're getting there. We can do glycomics, the release glycan complement of the cells. So that's, we take the sugars off, whatever their carriers are. And mass spectrometry is obviously the main tool in our science at this stage. There's glycoproteins, so you'll know lots of single proteins or simple mixtures of recombinant proteins are now relatively straightforward to characterize the glycosylation. Glycoproteomics, which is looking at complex mixtures of glycoproteins, so from a whole cell lysate or a whole tissue, etc., is still not there, but I'll talk about that in a minute. And of course, then I've grayed out the ones, proteoglycans, glycoinformatics, imaging, glycolipids. They're there, they're coming, their technology is being developed. But what is missing for the world to take notice of these molecules is what do they do? And I think I'd like to say that the reason that it had been so slow in reaching the forefront there is that there is no single thing that a glyco conjugate does, or the addition. It's not like a phosphate or a single ubiquitin or something which modifies it. We can have as many examples as you can think of. And this is some of the examples. Obviously, the first one we always use in lectures is our ABO blood group, is sugars. Single change in a sugar changes you from an A to a B blood group, and all know the ramifications of changing blood group. These antigens can be on proteins, they can also be on lipids. IgG has probably been the most looked at glycoprotein in terms of structure and function. A single fucose in this region here has been shown to change the complement, downstream complement activation by 100-fold. Reproduction, the reproductive tract is which is glycosylated and controls how the sperm goes up. That's pretty important. Microbiome interactions are suddenly coming into the forefront, where the bacteria that populate our gut are being found to be increasingly correlated with the mucus laying and the sugars that line the gut. Virus influenza has been for a long time known to recognize specific sugar linkages on the surface of our cells and gain entry there. So there's lots of functions that have been there. This one here is HIV. It's covered by glycosylation. One of the reasons it's difficult to get a vaccine against it. And of course, I can't go without showing the SARS-CoV spike protein. This is a beautiful picture showing the effect of the glycosylation on the accessibility of the sites on the virus. The purple and green things, right, are all the hairy sugars on the SARS protein. So this is a slide from Jerry Hart, which really just lists every human disease that we know of at this stage that involves glycosylation: inflammation, immune response, cancer, of course, infectious disease, Alzheimer's, diabetes, drug interactions, cardiovascular disease, genetic diseases. We can go on and on and on. So I think this session that we're doing for the HPP is highly relevant in the disease state, at least as far as I see it. Cancer has been known for years, decades, probably older than I am, that there is a glycosylation change able to be picked up in just about every cancer that you can think of, which is not surprising because the surface of the cell has to change to metastasize. And the genes, the glycosyltransferase genes, have now been isolated from just about every part of the synthetic pathway that are affected in the cancer process. So let's go to the technology, Glycomics. We can just about do that. And sometimes we don't even need to know what's carrying it. So is it a protein? Is it a lipid? It sometimes does not even matter. Sometimes it does, clearly. And we need to know how the sugar interacts with the protein. But sometimes it's just that surface that's exposed to the environment. And we can do Glycomics, which is release glycans, almost routinely now. It's just a standard mass spectrometry that we do in our lab and other people do in theirs. We prefer the chromatographic separation on graphitized carbon, which you can see here separates isomers, i.e. sugar structures which have exactly the same mass, but separate chromatographically. So we like that because we get a really good idea of how the epitopes, how the glycans are expressed in the environment, and single linkage changes can change the receptor that recognizes it. So this is a salivary N glycans that are pretty complex. the O's are just as complex. So, but we're still not there. We do miss structures unless we look for them specifically in mass spectrometry. Polysialic acid is too big. We don't see it when we do classic chromatography and mass spectrometry. On the other end of the size, we have the Porthy-Mannose structures, which are those which are, as you can see here, have been synthesized. They're quite small. This is work that's also done in Australia by Morton. Ed is concentrating on the polysialic acid separation. So we're trying to expand our repertoire and therefore look at the function of things we didn't know were there and we are seeing are important in disease. On the other hand, glycoproteomics, are we there yet is one of the things I asked, and we are largely there. We're not completely there. So we do it at the glycopeptide level. We can see the compositions of the sugars. We can see where they're attached to the protein. We can identify the protein. We can get some idea of the molar distribution on the different sites. But we are still, and we can now, by derivatizations, have a look at what sialic acid is there, modifications, but we are still missing some things. Quite difficult to see a lot of the linkage types in the structures, which in our view are really important. You know, whether you've got a 2,3 or a 2,6 sialic acid is completely different when you look at a receptor. Whether you've got a bisecting GLUTNAC or a terminal GLUTNAC, it's completely different recognition. So the linkage of these monosaccharides we think is really important. The molar site occupancy is quite hard to obtain. And then the branching of the structures, whether it's a triantennary or whether it's a polylactosamine chain on a biantennary, is still difficult to determine. So we're not quite there yet. But the sequence that we use is actually, we call it Glycomics-assisted glycoproteomics. So by taking off the sugars and doing Glycomics, so just, this is N-links, but PNGSF, doing the Glycomics, we can then also do D-Glycoproteomics, which is basically identifying where the sugars have come from. So the sites, the deamidation sites in the peptides that are left. And then we do the trypsin digest and helic enrichment, fractionated in chromatography, and put it together with use the Glycomics we found, the sites we found, to try and determine which peptides have which sugars on them in which protein. So that the technology is there. Doesn't answer everything, but hey. Okay, so let's move on to functions, which is really the topic of this seminar. And they're the sort of areas that we concentrate on at the moment in our lab. So there's microbial adhesion, which is through the sugars, the immune regulation. We're doing imaging by mass spectrometry and lectins, etc. We're finding diagnostic biomarkers. And I sat there over the weekend and I thought, I have no idea which one of these I'm going to talk about. So at the end of the day, I decided to talk about the microbial adhesion function of sugars, because that's something that I've been interested in probably for most of my career. So I'll give you one example in which is the group A streptococcal or gas disease. It's a collaboration with Martina. A lot of the work's done by Arun and David at the University of Wollongong in Australia and the Glycomics Institute in Queensland. And the gas, or the streptococcus, has two proteins called M3 and M12. They're lectins, and they significantly contribute to the infection process. So this lovely picture that David sent me is terrible. It shows terrible photographs of the streptococcus infection of our skin and throat, and also on our hands. It can lead to secondary rheumatic heart disease. It kills people, and it's quite common in our Indigenous communities. So that's one topic of that the WHO has given a priority for vaccine. Now, needless to say, these proteins actually have a binding affinity for the ABO blood group. So if you take a recombinant protein, do SPR, this is the M3 and the M12. The M3 is the orange and the M12 is the blue. You can see as you go up with the blood group A, blood group B, blood group H, you can see an increased binding of these proteins to these blood group red blood cells. The ultimate thing is to go into people and see whether it affects their tendency for infection, or does it infect well, the rate of attachment. So we got saliva of healthy donors, analyzed them, and also collected the buccal cells and used the buccal cells to examine how these bacterial proteins bind to the surface antigens. So different blood groups plus non-secretors, they're people who do not express a blood group, which is shown over here. Can you see my arrow on the screen? Yeah, okay, thank you. And so we looked at blood group antigen status and the M3, M12 association to the epithelial cells. So this is just the lining of the mouth. It was just scraped out from healthy donors. And you can see if you look at the blood groups here, the binding of both the M3 and serotype of the M12 changes. This is flow cytometry, and you can see A and H, or blood group O, have stronger binding than blood group B, which has a weaker binding to both of those proteins. And then we played with it a bit and treated it with exoglycosidases of the buccal cells and see what happens when you change the sugar profile of those cells. Now in this case, it's again M3 and M12 protein. We took off fucose, just with a fucosidase, and measured the binding of these proteins to the different blood groups. And you can see if you take off fucose, you change the relative binding. This is your control over here. And you take off fucose and it primarily still goes to B, but the others have dropped. But the M12 doesn't seem to care very much. If you take off galactose, it stops the binding of both the M3 and the less so the M12, but completely knocks out the M3 binding. And sialic acid, which is not a part of the blood group, but you take off sialic acid and it decreases the colonization of all the blood types. So it's just, I thought I'd use it as an example to say bacteria bind to the sugars. They determine, and I also thought it'd tie in nicely to Stacy's talk later, because most of these sugars are expressed on the mucus in the mouth, as well as the other proteins, but they, the blood groups are expressed also on the mucus. So then you say, so what? We know that they bind to the sugars, but can we use this fact in a therapeutic way? So what we did was just pull saliva, take off the end links in this case, and just use them in the assay to see if they would do anything. So what you can see here is in the binding, this is just straight the association of the M3 with the saliva. If you pre-mix the bacteria with the salivary glycans, you reduce the binding of the bacteria to the cells, i.e. that you've got competitive binding of the saliva with the bacteria for the binding sites on the buccal cells. So this inhibited the colonization of the bacteria on the buccal cells. So you can imagine carbohydrate-based therapeutics in the future, in which you have a surface here, you've got your carbohydrates there, you've got your mucus sitting above the cell surface, usually it's lining the surface, and you get some sugar mimics that you just wash your mouth out. And that didn't work very well, but never mind. You just wash your mouth out with the sugar mimics, wash your mouth out. It competitively binds to the bacteria and stops it binding to the surface of the cells. So just to finish off for this setting here, I wanted to show you the rise of glycoproteomics over the years. This is the process of publications in the field of glycoproteomics, the analysis of the glycosylation of proteins and the glycopeptide analysis. It's gone incredibly up from, well, this is the glycosylated peptides reported. You're looking at 2005 starting, and then now it's going through the roof. This was in a review in 2016. Further review in 2020 showed that it's increasing exponentially from single experiments. So the world is paying attention. Are we there yet? Not completely in glycoproteomics. You can see that we can identify what's carrying it. We can determine the glycosylation site in most things, but the site of occupancy is still hard to do in complex mixtures, as is the topology, the linkages, the branch points, and the relative abundance. So tertiary structure is another world, and multiple glycosylation sites in one peptide are also difficult. But hey, we're getting lots of information now. We're getting lots of knowledge. And the drawback, I believe, is that we need better software to do this. Proteomics took off in the way it did when the software caught up with being able to analyze the data in a high throughput manner and a reliable, accurate manner. So I think that is a shortcoming at the moment in our field. The HPP, biology disease HPP, Morten and I are chairing the Human Glycoproteomics Initiative in that area. It used to be called the Glycomics Proteome Initiative. We gave it a little bit of a flick over to a different name, included Glycoproteomics. We've just done our first study, which is an interlaboratory comparison of the mass spectrometry analysis software available both commercially and academically to the community. It was a huge endeavor, and we have now had, it's on bio-RIV, but it's under review in Nature Methods at the moment. So hopefully it will be published shortly. In addition to that, we've formed an alliance to try and internationalize glycoinformatics. It's a consortium we're calling the GlySpace Alliance, which involves the Glycosmos group in Japan, the Glygen group in USA, and the Glycomics at Expazi group in Switzerland. And what we're trying to do there is work together to unify the software, the nomenclature, the data standards, etc., the metadata that's collected, to try and link all the information out there on glycosylation of proteins. So if you want to do glycoproteomics well, our conclusion in that paper was that you need a powerful search engine. You need to set the search settings carefully and bring in post-search filtering and know something about your glycobiology. I'm going to do a last plug. I think I'm at time. We are having the 3rd Australasian Symposium, Glycoscience Symposium, on the 3rd or 4th of June. As you can imagine, it'll be online. We started this in Australia. This year it's going to be in Japan, and we have a pretty starry confirmed list of speakers, and I didn't point at myself then, from Asia, Australia, India, and it should be really interesting. So if anyone wants to join up, please feel free. And thank you to my group. I'm at 2 universities, one in Queensland and one in New South Wales. And we thank all our sponsors. Thank you very much.
Thank you very much, Nicki. That was a really, really, really long, exciting journey. No, really. I found a lot of questions in the question and answer corner. So maybe Ileana, how can we give some?
So we are quite a bit over time, but maybe let's take a few questions and maybe a few minutes of questions, and then we keep some of the remaining questions for the panel discussion at the end.
Okay, so I pick the first three. So it's just the first. How many current drugs are targeted to glycoproteins?
How long is a piece of string? Ed, that's not a nice one. I don't know. I mean, things like CA125, etc., have been used as diagnostics for years. CA19-9. Drugs targeted? I don't know. Maybe somebody else can answer that. That's a good question. Probably none. None that I can recall off the top of my head. Does anyone else have an idea of a drug that is? Oh, of course. I mean, there's the influenza drug. There's, you know, the what's it called? Tamiflu and what's it called? Come on, it's an Australian drug that interferes with the binding of the influenza virus to the cells and stops the virus being released from the cells. Mark von Instein will not talk to me anymore, seeing he developed it.
Important in the infectious disease. I mean, in line with what you showed in the slides.
Yes.
Before, so it's
Yes.
really fundamental, the link. I think another one, and then we move on. Ileana, is okay. Hi, Nicki, you mentioned the potential number of glycosylation sites and isoforms, but the largest study of N-linked glycopeptide characterization has not identified more than a few thousand glycopeptide isoforms on a few hundreds N-linked sites. Why is that so?
Okay, first I'm going to say that I was just sent a text because I've obviously at midnight, my memory has gone. It's called Relenza is the drug I'm thinking of, which is the, sorry, that was from before. Thank you, Morten. All right, that's why haven't we got more information? I would say two reasons, or many reasons. One, we haven't looked enough. Two, the glycosylation sites are easy to find by classic proteomics methods. Glycopeptide isoforms, you have to actually go enrich the glycopeptides, separate, if possible, as much as possible, the isoforms, do HCD, CID, ETD types of mass spectrometry to get the different masses that are on those isoforms. So it's heavy mass spectrometry, and it needs the software to make it easier.
Thank you very much, Nicki. I think for time reason, we probably have to unfortunately skip the many other questions received. I mean, maybe you can chat them directly. We use them we address them in the discussion at the end of the presentations.
Sure.
Many thanks again.
Thank you.
Hello, everyone.
Alexandra. Yes, Alexandra.
Yes, my name is Alexandra Nita-Lazar, and on behalf of the D-HPP committee and the webinar organizers, I would like to introduce the next speaker, Professor Boris Macek from the University of Tübingen. Boris actually has experience in glycosylation too. He did his PhD with Jasna Peter Katalinic in Münster. And then he did his postdoc with Matthias Mann, so moved on to other exciting modifications, especially phosphorylation, first in Denmark and then back in Germany, where he remained for his independent lab, moving just to Tübingen in 2008, where he leads his lab of quantitative proteomics as well as the proteomics core center. And Boris is a pioneer and an expert in bacterial phosphorylation and kinases, and this is the topic of his talk today. So Boris, please.
So thanks a lot, Ola. Thanks for your invitation, and I'm really happy to be here. And you mentioned my brief history in glycobiology, and I'm particularly honored to speak after Professor Packer, who was my big hero 20 years back when I worked in Jasna's lab. And since I moved to more like signaling and phosphorylation, which is much easier to do than any glycobiology, I must say. So I thought for this particular webinar, it would be also good to look into pathogens themselves and also raise awareness that bacteria also have a myriad of modifications. So this is from a recent review that we published. So, and you know, many of these modifications will be actually very familiar, even to cancer biologists, right? So serine, threonine, tyrosine phosphorylation is not common in bacteria, but some prominent pathogens do actually have this kind of modification. You will see a lot of you know, lysine acetylation. You will see ubiquitin-like modifications. You will obviously also see glycosylation and so on. So we're really interested in these modifications, and I'm actually particularly interested in serine, threonine, and tyrosine phosphorylation in bacteria. And so obviously here you see, you know, these modifications are involved in many functions. So there is no like unified, you know, function that you can now just say, and we're basically just trying to explore those. And what really, you know, took our attention in the past years is this process of persistence in bacteria. So I have to actually introduce it a bit. So all of us are aware of resistance mechanisms. However, in addition to resistance, some pathogens can actually survive antibiotic treatments by shutting down their metabolism. So they pretty much go to sleep. They're still sensitive to antibiotics, but very few cells that actually survive the initial treatment, and this is seen in this killing curve here, very few of these cells actually survive this treatment because they shut down their metabolism. So this is a very you know, relevant biomedical issue, because you know, many of these pathogens like, you know, Klebsiella, also E. coli, in pathogenic variants, that actually can persist, right? So what really rose our interest in that field is that this process is thought to be controlled by toxin antitoxin systems, and one prominent system would be so-called HipA system. And interestingly, this HipA is a kinase. This is a serine threonine kinase, so very rare actually in you know, bacterial genomes. I think E. coli will have not more than 4 or 5 of these. And this particular kinase actually phosphorylates a very, you know, prominent target, which is glutamate tRNA synthetase. And by phosphorylation of this glutamate tRNA synthesis, it's thought that basically this triggers, actually I should probably move this somewhere else. So it triggers a process that basically leads to, you know, inhibition of translation, a lot of uncharged tRNA floating in the cell, and then triggering downstream responses like stringent response, production of this PP, GPP, alarmone, and eventually it leads to the persistence. I must say that this whole process is pretty debated in the field of microbiology, but what rose our interest was really the fact that at least in this prominent case, and I should also say that HipA was the very first gene that was ever linked to the persistence in bacteria several decades ago, that this is actually encoding serine threonine kinase. I should here also mention that this is an operon where HipB is an antitoxin that binds HipA and inhibits the HipA, right? So basically it's not a phosphatase, but it's just, you know, structurally inhibits the HipA when everything is normal inside of the cell. So obviously this major substrate was discovered by others and published, but we reasoned that this is likely not the only substrate of this particular kinase. And actually, while reading these papers, we saw that there are a couple of things to be answered and inconsistencies in the literature. So for example, what happens when you overexpress HipA on a plasmid, you actually induce arrest. So the culture doesn't grow anymore. So it's a growth arrest, so it's a toxin really. and you increase the persistence, right? So the number of cells that will eventually be persistent. But there is a variant, it's called HipA7. So it's the same kinase, it has 2-point mutations, right? You overexpress this one, right? And you do not have this growth arrest, and you do have many more persisters, right? So this was a very unclear, feature in the field. So we actually thought maybe this is governed by the substrates of this kinase, or I should say of these two forms of the kinase. So we actually teamed up with Ken Gaddis, who worked intensively on these at that time. And we actually designed what from proteomics point of view is a very simple experiment. You would use exactly this model. You would overexpress HipA in basically wild type E. coli cells, and you will monitor the phosphoproteome over time, right? So these are 3 points. We actually employ Silac in this experiment. So Silac, many of you know from eukaryotic cultures. So we actually routinely also employ this in prokaryotic, especially E. coli cultures. So we can compare 3 states. In this case, then we basically lyse the cells. We could combine those cell extracts, and we digested the proteome with lyse-C and with chymotrypsin. It was important because we wanted to find GLTX, which then would not be accessible with lyse-C only as the protease. So you use a combination of 2 proteases, and then you run a standard PTM workflow based on titanium oxide enrichment of phosphopeptides. And then in parallel, you also need to check the proteome, so the protein levels, because you need to normalize this phosphodata for the protein levels in order to see potential kinase substrates, right? You measure all of this. We use Orbitrap in our lab, and then using Max Quant, we basically analyze the data. So it's pretty much a routine for every phosphoproteomics lab. And in addition, we also designed the experiment in which, again, in a triple Silac fashion, we directly compared the HipA7 and HipA variants of that particular kinase, just to see whether the the substrates are similar or not, right? So, and these are the results. We published this a few years back. So basically, this is how the growth curve looks like. You have 2 plasmids, right, in the cell, and you can induce the first one, HipA, with arabinose, and you kind of induce this growth arrest. And then what happens here, and this is what we can see here. So every dot here is a phosphopeptide, and obviously it's a log2 scale. So 1 to 1 is no change, and you see that there is actually several several dozens of substrate that actually respond to overexpression of HipA. And nicely, one of the major substrates we see is the GLTX, was known before, so it's our positive control. And now there are several interesting substrates in addition to that. And by overexpressing HipB a few hours after that, we actually then said those that are not direct substrates of the HipA, they will remain, but this is what is very likely to be direct substrate that actually should go back and decrease in abundance after we overexpress the antitoxin, right? And this is exactly what happened. So for several of those that we detected phosphorylation sites, we saw this particular pattern, and we further then validated them, for example, with in vitro kinase assays. And so Maya Szymański, who was really the person in charge, also did a lot of additional follow-up, which is all published, and you can read it. But for in the interest of time, I cannot go into all details. Now, this was a very interesting experiment. I said we also designed an experiment when we directly compared these 2 kinases, or these 2 kinase variants. And interestingly, this is seen here, when we actually overexpressed HipA7, right? We actually thought we would see more substrates because, we expected more persisters, but quite on the contrary, we saw less. We did see this main substrate, so GLTX, we did confirm that HipA7 actually phosphorylates this as a main target, which was not known before, but we didn't see much more. And this is the direct comparison. This is an elegant screen in which you can actually compare the activity of two kinases or two variants, right? And you see that both of them will phosphorylate GLTX. On this side, we see HipA with numerous substrates, and we see HipA7 on this side with pretty much no substrates, right? So this was puzzling. We did confirm all of this through in vitro kinase assays, and you see here HipA when incubated with GLTX, we need to add tRNA in order to get GLTX in proper conformation. You see a lot of activity which you do not see when you have HipA7 under the same conditions. So HipA7 is indeed a weaker kinase, right? Contrary to what many people have expected. And this is also confirmed by looking at autophosphorylation sites of these two kinases. So basically there is an autophosphorylation site that you can also check, and we see it less phosphorylated in HipA7. And then finally, we basically designed the experiment in which we expressed those two kinases from the chromosome. So it was not overexpression, it was really from the chromosome. And the HipA is basically so toxic under these conditions that you don't really get viable cells. But we did get results for HipA7, again confirming that there is very few. I mean, we see the GLTX, we see this PSPA, which is likely a substrate, but this we actually never really checked, so one will need to validate that. So this is still a confirmation that we get similar results even when we express those from the chromosome. So the theory is now, why do we see such difference in the activities of those? Or actually in the phenotype, right, is likely because these 2 point mutations that HipA7 has prevent the binding of HipB. So HipA7, even though it's a weaker kinase, it never gets really inhibited by HipB. So it can actually persist in the cell, even despite of the HipB expression. And this is not the case with HipA. HipA is a stronger kinase, but it is very efficiently inhibited by HipB. So we postulate, we didn't prove that, but we postulate that this must be the reason for these phenotypes, right? Now, Ola, at this point, you'll tell me how many minutes I still have?
We are so much over time that
I will actually cut it a bit short, because just wanted to say that we now are following up this HipA biology by actually measuring protein turnover. So this HipA, HipB system is a very nice system in which you can induce persistence, and you can then wake up the cells, right, by overexpressing HipB, right? So and what you can then do, you can do something that many of you know, dynamic style, you can actually pulse a stable isotope labeled amino acid in the culture as you, for example, wake up the cells, and you can measure incorporation of that particular amino acids into the protein in order to measure newly synthesized proteins. And this is exactly something that we now have submitted. And I will now cut this part because it's not strictly connected to modifications, but it is triggered by a kinase, right? I just wanted to make, to finish then with this, what we can actually measure with this are newly synthesized proteins. And what interestingly, we see a lot of, over 1,000 proteins that we can actually monitor in that process. But if we look into the ribosome, we actually see how, if you look at this blue and yellow, how specific ribosomal proteins are rearranging, right? So we're here now entering the stationary phase of the bacterial growth, and you see how basically two paralogs are, changing abundances and exchanging their places in the ribosome. And I'm mentioning this is super interesting. People saw it before. Now we see it with this dynamic stylet. But interestingly, this particular protein is also phosphorylated, right? Not by HipA, but this one of the other kinases that I mentioned in E. coli So we actually see a strong link between general translation in bacteria and phosphorylation. So this is something that we now are investigating in the lab. And so obviously this is just an outlook. HipA is not an isolated case. A lot of proteobacteria do have this particular operon, and we're now investigating some of them. Actually, in many cases, there are still uncharacterized operons, for example, in Klebsiella and so on. And so we are now investigating the role of these HipA-like operons in these pathogens and persistence of these pathogens. Now, with this, in the interest of time, I will not really reach the conclusions. I guess you got my point. We have very robust methods at hand to check protein phosphorylation, even in bacteria. And I really do hope that this will lead to some very, very important biomedical findings and basically new drug targets, obviously. So in the last slide, I just want to thank especially Maya, who I mentioned already. Fabio, it was a typo, sorry, continued her project, and Payal is currently a PhD student in the group who is also working on some aspects of the project, and Ken was our main collaborator in this. These are our funding agencies, and with this, I will thank you for your attention.
Thank you very much, Boris. And I will start with the questions. If there will be a lot of questions, then we will move them again to the panel. But I see the first question from Dr. Tao in the chat. Boris, does high toxicity of overexpressing HipA7 or other kinases come from the fact that there are few phosphatases in the bacteria to balance the signaling?
Yeah, this could be. So first of all, you cannot really generalize. So some bacteria have numerous kinases, serine-threonine kinases, and also this eukaryotic-like kinases. I don't really like this term because actually those kinases were even present in bacteria before eukaryotes, so evolutionary in evolutionary terms. Some of them also have many phosphatases. So, for example, cyanobacteria, they also have, a relatively high number. It's never compared to eukaryotes, always less than eukaryotes, but several, up to 10 in the genome phosphatases as well. And it could well be that this toxicity, is there because there is nothing that removes this particular modification, but we just don't know. What I actually think that in that particular case, the toxicity is more connected to the substrates themselves, because several of them are really involved in, very, very essential processes of especially translation.
And another question in the Q&A from Thomas, a technical question. In your volcano plots, you show a score for each phosphopeptide. Can you explain again how you calculate the score, and why do you use log 10 of the p-value and not the adjusted p-value?
Oh, okay. So that's now a good question. I mean, this experiment, I will not go back into the details, but obviously we would be doing, a t-test and, a standard volcano plot. Now, why log 10 and not, then I would really need to go back and look into that. But it was obviously, one challenge there is also that you don't have that many data points to begin with, right? So all of those, bacterial phosphoproteome experiments are also leaving you with relatively few substrates, right? So I would need to really look into the details why not one way or the other, but I think that doesn't really influence the results.
All right, I have also a lot of questions, but let's wait with more questions to the panel discussion. Thank you very much, Boris, again, and let's move on to the next speaker.
So it's my honor to introduce the next speaker. So our next speaker is Dr. Claire Eyers from the University of Liverpool. Dr. Eyers is the professor of biological mass spectrometry and the director of the Center for Proteome Research at the University of Liverpool. Her research focuses on developing mass spectrometry and ion mobility techniques to study signal transduction in the cell. And her group has published multiple influential works on the role of protein phosphorylation through various protein kinases in driving disease onset and progression. And the title of her talk today is Phosphorylation Mediated Signaling, a New Era. Thank you.
Thanks. Sorry, I couldn't find my unmute button. Thanks very much, Maggie and Liliana, for the invitation. Yes, so I'm going to be talking about phosphorylation, but unlike Boris's talk, my focus is entirely on mammalian, on vertebrate signaling. Now, I'm sure that Nicki and I could talk well into the late hours about which is more important, phosphorylation or glycosylation, and I guess dependent on what day you caught me on, I could argue either way. Right now, though, I'm going to argue for the fact that phosphorylation is largely the more important of the two post-translational modifications. And that is because of the diversity of the things. I know Nicki talked about the whole host of diseases that glycosylation can affect. Phosphorylation is very similar. We have a whole host of physiological responses and effects that are regulated by protein phosphorylation. And that is because of the different types of protein processes that phosphorylation can regulate. So we're talking about changes in catalytic activity, both positive and negative. Lots of demonstrations where we change the binding and the interactions with various biomolecules as a result of phosphorylation. Stability, both directly and indirectly through differential ubiquitination, for example, as a result of phosphorylation, and altered subcellular localization. So when we think about vertebrate or human phosphorylation, we think about serine, threonine, and tyrosine. So in the same way that Boris talked about serine and threonine phosphorylation in bacteria, this is really what we think about all the time in terms of human phosphorylation, with the vast majority of phosphate occurring on serine residues. But in actual fact, we have a whole host of other residues which can be phosphorylated. Histidine can be phosphorylated. Actually, it forms two phosphoisomers dependent on where that phosphate group is on the imidazole ring. So you can either have one phosphohistidine or three phosphohistidine. You can also have phospholysine, phosphoarginine, which Boris also mentioned, phosphospartate, phosphoglutamate, and phosphocysteine. Yet all of our studies in vertebrate systems have focused entirely on these three. So the question really is, in humans, in other mammalian systems, how much do these different types of phosphorylation actually really exist? So we've been interested in this question probably for about the last 10 or so years. And as Maggie said, we spent a number of years doing phosphoproteomics, both on targeted proteins of interest, but also high throughput phosphoproteomics. And the standard workflow, which the previous speaker touched on, really involves phosphopeptide enrichment using iMac or titanium dioxide preferentially. So you would take your cell lysate, you would digest that with your protease of choice. It's typically trypsin or lyc, generating a mixture of phosphorylated and non-phosphorylated peptides. You would then enrich for those phosphopeptides to kind of strengthen your pool, and then subject that to reverse phase tandem mass spectrometry. And if we're thinking about serine, threonine, and tyrosine phosphorylation, these are phosphomonoesters, which is great because they're stable. This phosphate group is stable under the low pH conditions required for optimal enrichment using iMac or titanium dioxide. When we consider atypical phosphorylation, so I've shown phosphohistidine in an example here, this phosphoamidate bond that you have is unstable at low pH. So this was really the driving force behind this project. We were interested in trying to develop a strategy for enriching for phosphohistidine in a manner that would maintain its stability for identification of phosphohistidine sites by reverse phase chromatography. Because if you consider the pH at which we would typically do a titanium dioxide enrichment at, that phosphoamidate bond is no longer stable. So how can you enrich for atypical phosphopeptides for site identification? So the method that we eventually came up with, and I'm summarizing in one slide here, an accumulation of probably about 5 or 6 years work, is something called UPAX, which stands for strong anion exchange chromatography for unbiased phosphopeptide enrichment. And it works under very simple principles, actually very similar to that which we use for titanium dioxide, namely electrostatic binding. So if you have an unmodified threonine, for example, that is then phosphorylated, you end up with a drop in your pKa to about 2.1. And that change in pKa is the same irrespective of whether or not you're talking about threonine phosphorylation or histidine phosphorylation when you induce that net negative charge. So the method that we developed, which we validated, or sorry, I should say that we developed the method using a mixture of serine and threonine phosphorylated casein peptides, and then a standard which we generated using phosphoamidate mediated phosphorylation of myoglobin, which we then characterize as containing 6 sites of histidine phosphorylation. Then uses strong anion exchange separation of those mixtures on a poly-Sachs column. And what we showed here is that the vast majority of the phosphopeptides from this mixture will elute in the latter fractions from this strong anion exchange chromatography system when we undertake this separation at pH 6.8, as you can see here in blue. And the vast majority of the non-phosphorylated peptides from this mixture come out in the early fractions. So phosphopeptides exhibit stronger interactions with the strong anion exchange than non-phosphopeptides, which allows us to separate the vast majority of our phosphopeptides from our non-phosphorylated peptide cohort. One thing that's worth pointing out here, this is not the same as doing a titanium dioxide or IMAC-based enrichment, where you can get, upwards of 95% phosphopeptide recovery, but it is a very good way of being able to enrich for certain pools of your strong anion exchange fractionation for things that are preferentially phosphorylated. Okay, so having developed this method, we want to see really how many sites of atypical phosphorylation we could see. So we took HeLa cells, we subjected them to our UPAX protocol, Sachs fractionation at pH 6.8, collecting 16 fractions, did a 2-hour LCMS gradient where we were using HCD fragmentation, so this was done on an Orbitrap fusion instrument, and then we used neutral loss of phosphoric acid to then trigger ETHCD. And in terms of data analysis, we did this using a mascot peptide identification using PTMRS to give us an indication of phosphosite localization. So as I said, when we first started this project, our focus really was on phosphohistidine, and that was because there'd been increasing evidence and discussion in the literature about putative physiological roles of histidine, and that was really what was the driving force behind this project. And when we looked at the number of phosphohistidine phosphorylation sites that we identified, and this is a relatively conservative PTMRS score of 0.90, we can see actually that the numbers we're identifying are not that dissimilar to the numbers that we were seeing for phosphotyrosine, possibly slightly lower. But as we started looking into this a little bit further and we started evaluating other sites of non-canonical phosphorylation or atypical phosphorylation, actually, with the exception of phosphocysteine, where the numbers are relatively low, all the different types of phosphate that we were looking for, actually the numbers are higher than we see for phosphotyrosine. One of the things that concerned us really when we started doing this type of analysis and looking for phosphorylation of all these different residues was whether or not this was an artifact of the fact that, as well as searching for variable modification phosphorylation of 3 residues, we're now actually looking for variable phosphorylation of 9 sites. So whether or not this is actually a consequence of the change in our bioinformatics procedure. So we actually took exactly the same cell lysate, we subjected it to titanium dioxide based phosphopeptide enrichment, and as a comparison of our UPAX strategy, searching both pools against all sites of phosphorylation. And if you look at this graph here, what I'm showing you is the proportion of phosphorylation sites that we identified, unique phosphorylation sites, where the grey sites represent those canonical sites of phosphorylation, and the coloured sites are the atypical sites of phosphorylation. And what you can see is that the number of sites we see and the proportion of the sites that we identify as atypical phosphorylation isn't purely a function of the fact that we're now searching for all these different sites of phosphorylation. Okay. So what can we do with this data? So we now have a data set where we have identified lots of different sites of atypical phosphorylation. And what this allowed us to do is a few things. We could start to look for consensus where we see sites of, for example, phosphohistidine. The data I'm showing you here is actually with a higher site localization confidence. So this is now at a PTMRS score of 0.99, which we thought more appropriate to do motif analysis on. And so you can see motif analysis for phosphohistidine. You can see that we have a preference for a hydrophobic residue immediately C-terminal to that site of phosphorylation, with a preference for either leucine, isoleucine, or valine, which we actually saw in over half of all the sites that we identified. We did the same thing for phosphoaspartate, where we showed that we have a preference for either proline, isoleucine, or leucine, and this was seen in about 40% of phosphorylation sites. One of the other things that we can do, as well as looking at motif, is to try and get a feeling for what type of proteins we see these atypical phosphorylation events on. So we see phosphoprotein coming up in this, which isn't really surprising because a lot of the times when we see phosphorylation, we also see other types of post-translational modification. The thing that was interesting for phosphoaspartate from our point of view are poly-A RNA binding proteins, and the fact that a lot of the proteins that we're seeing seem to be in the nucleoplasm or the nucleus. With phosphoglutamate, we see a very similar type of GO profile when we start to look at the type of proteins that we see phosphoglutamate phosphorylation on. But actually the motif analysis is different. So rather than having something where we see proline, leucine, or isoleucine at position +1, actually we seem to see two motifs, one which either is a basic consensus with a lysine at -6 or +7, or with an acidic consensus. So between these two, it implies actually that there may be a role for phosphoaspartate and phosphoglutamate in transcriptional control, but we've yet to start digging into this data anymore to start validating some of these effects. Phospholysine, we can, using motif analysis, see a preference for lysine lysine at -5. Phosphoarginine, we can see a glycine at +1. Phosphocystine, actually the sites of phosphocystine we saw there were not very many at this PTMRS score of 0.99, but actually this turns out to be one of the more interesting features of some of the data that we identified. So that's all of really the proteomics data in terms of the discovery study. I'm just going to move on now into really starting to explore what we think this data means. So the issue for us is that we found all these phosphorylation sites, but to our knowledge, actually, there is no enzyme that will regulate phosphorylation of atypical phosphorylation in human cells. So I think Boris touched on this earlier. For bacterial signaling, there's two component systems, for example, where you will get phosphorylation of histidine and then relay from phosphohistidine to phosphoaspartate. But there's no such system that really exists that is known to be functional in mammalian cells. So the question really is, what is the enzyme, or what are the enzymes that are regulating these atypical phosphorylation events? Now, we hypothesized that some of these atypical phosphorylation events may be autocatalytic, and that is because a lot of protein kinases are known to autophosphorylate. So we screened the data for all the protein kinases that contain sites of atypical phosphorylation, particularly where those sites were in the catalytic domain. So you can see some of that data here. What I'm going to focus on for the rest of today's talk is a protein called PINK1, which is known to be involved in Parkinson's disease. So PINK1 was phosphorylated within the DFG and the APE motifs. You can see the APE motifs here on a phosphocysteine residue. And the reason this motif is important because this is the activation segment. So this region between DFG and APE, phosphorylation is required for activation of all kinases, including PINK1. So we wanted to validate, firstly, whether or not we could replicate phosphorylation of this cysteine residue, and also to try and figure out what phosphorylation of that residue did. So we started a collaboration with Marathel Muckwit in the University of Dundee in Scotland, who's been working in the field of PINK1 and Parkinson signaling now for a number of years. So we expressed either a wild type PINK1 or a kinase inactive PINK1 in 293 cells. And we can induce expression of PINK1 using doxycycline and CCCP, which is a mitochondrial uncoupling agent. We then extracted the mitochondria, separated these by SDS page, and then subjected these to LCMS analysis. And what we did initially is confirm our site of cysteine phosphorylation on 412, which is brilliant. What was actually much more exciting is that this phosphorylation event appears to be an autocatalytic event because we only see this site of cysteine phosphorylation with wild type enzyme, but not with the catalytically inactive kinase dead protein kinase. So we think we've identified PINK1, therefore, as a cysteine protein kinase, which would make it the first human cysteine protein kinase to be identified. But of course, that raises a whole host of other questions. So are there other serine threonine protein kinases that double up as cysteine protein kinases? And also, if PINK1 is capable of phosphorylating cysteine in an autophosphorylation event, are there other PINK1 substrates? So then just briefly in terms of function, because this is a short talk. So we wanted to look at what we thought that cysteine phosphorylation might be doing. So we expressed both a phosphorylation null, so a cysteine to alanine mutant, and a phosphorylation mimetic. It's not really mimetic, but it's something that is putatively potential for being phosphorylated, so a cysteine to serine. And what we see is really interesting. We do not see a change, if I focus down here, in PINK1 autophosphorylation on serine 288, irrespective of that mutation. What we do see is elevated levels of ubiquitin serine phosphorylation. And this serine phosphorylation is known to be essential for downstream mitophagy. So with the cysteine 412 alanine, which is incapable of then being phosphorylated, we see an increased level of ubiquitin serine phosphorylation, suggesting that that is actually essential a residue for mitophagy. Okay, and just finally to say that this cysteine residue is really highly conserved. So we think this may be a very general effect. Okay. In the instance of time, I'm just going to skip this slide in detail just to say that these cysteine residues are highly conserved. So 33% of eukaryotic protein kinases have a cysteine at that exact same position. We also have identified two other motifs where we see cysteine residues which are known to be phosphorylated within this DFG activation loop of protein kinases. And what's really interesting, and we think this is where we start to see PTM interplay, is the fact that these cysteine residues in a number of enzymes we show both to be phosphorylated and redox modified, which suggests that there may be actually regulation or co-regulation between phosphorylation versus oxidation. Okay, so to summarize then, we've developed and shown that we can develop a method for enriching for atypical phosphorylation events as well as canonical serine threonine and tyrosine. We've shown that this is ubiquitous in human cells, and what I haven't shown here is actually these events are dynamic, and there are implications for aspartate and glutamate phosphorylation and transcriptional regulation. We've also shown that PINK1 autophosphorylates on a cysteine residue, and that regulates the activating ubiquitin phosphorylation event. And there appears to be interplay between cysteine phosphorylation and redox regulation within the protein kinase catalytic domain, which in turn regulates protein kinase function. But we still really don't know what the roles in regulation of the vast majority of the non-canonical phosphorylation sites are that we discovered. So this is really going to take quite a concerted action from our group and others. Finally, just to highlight Gemma Hardman, who developed this protocol, and Chris Clark, who's been continuing, and then Maratle, who's really been, his group has been essential to establishing a physiological role for PINK1. Thank you very much.
Okay, so we have a number of interesting questions. So I'm going to read them most sequentially. So the first one is, hi Claire, in all textbooks, it's mentioned that histidine phosphorylation is unstable at low pH. But how fast do you lose the phosphate group at lower pH? Have you done the incubation with several synthetic histidine phosphorylated peptides? I have seen examples where it takes hours to lose 50%, which do not exclude the conventional IMAC and titanium dioxide methods for enrichment of histidine phosphorylation, as it takes only minutes for the enrichment. Please elaborate.
Okay, so first thing I would say is it doesn't take only minutes for the enrichment. The second thing I would say is that yes, in both our bioarchive paper and the EMBO journal paper, we did do a time course analysis of phosphohistidine hydrolysis, as did Simone Lemire actually. And we both look at rate of hydrolysis at different pH values. In our study, what we show is that the half-life of hydrolysis of phosphohistidine is context dependent. So it depends on the amino acids surrounding that site of histidine phosphorylation. But the half-life can be as short as about 15 minutes when you look down at about pH 2, which is really what you need for titanium dioxide enrichment.
Okay. So the second question, I think it's slightly similar. So nice sample preparation approach. However, how do you account for the acidic conditions during the LC-MS separation? Could significant degradation happen in that time frame? What alternatives would be available for chromatographic separation and ionization?
Okay, so we have actually tweaked our LC-MS conditions. So we load onto our LC-MS column in water, so our samples are not acidified before they go onto the column. When we do offline desalting, we also do not add acid to those offline desalting steps. By the time that you bound it to your reverse phase column, yes, we will be getting hydrolysis. The rate of hydrolysis will be lower because at that point it is bound to solid phase resin. So the kinetics will say that the rate of hydrolysis is lower. But it's a hit we have to take. We spent a number of years trying to optimize ammonium acetate based neutral pH for doing online LC-MS, and for a variety of reasons, we elected not to carry on with that.
Okay, so in the interest of time, I'm going to read out one more question. So which fragmentation method have you used for identifying the cysteine phosphorylation in PINK1?
We've done a number. We've done HCD. We've also done ETHCD.
Okay, so thanks very, very much, Claire, for the wonderful talk. So I am now going to move on to the next speaker. And our next speaker is Dr. Laura Murray-Nerger, who graduated from Dr. Ileana Cristia's laboratory in Princeton, where she aimed to uncover how nuclear protein-protein interactions that are modulated by acetylation play antiviral roles during infection. So Dr. Murray-Nerger is currently a postdoctoral scientist at Harvard Medical School. In her presentation, Laura will talk to us about how acetylation of the nuclear lamina regulates nuclear periphery stability and function. Thank you.
Thank you so much for that introduction, and thank you for the opportunity to share my work today. In line with some of the other PTMs that have been discussed today, I think it will come as no surprise that acetylation also functions as a really important regulator of many cellular processes in homeostatic cells. And acetylation can have diverse roles with regard to regulating enzymatic function, protein-protein interactions, and protein localization, among many other functions. But something that's becoming more and more apparent in recent years is that with studies of specific proteins during infection, protein acetylation can function as a regulatory toggle during viral infection. So for example, viral genomes that can become chromatinized can have their gene expression regulated by acetylation of histones, but also acetylation of either host or viral transcription factors can mediate their functions. Additionally, our lab and some other labs have shown that acetylation of the DNA sensor I516 can dictate its subcellular localization during infection. And finally, acetylation, of course, is not constrained to the nucleus, and many labs have shown that acetylation of microtubules is really important for stabilizing them, which can facilitate trafficking of viral proteins and viral particles themselves during infection. However, when we were thinking about this, we realized that there was really limited understanding on a broad scale of how protein acetylation may serve as a regulatory toggle during viral infection. And so we wanted to ask first, in a temporal and spatial manner, how is protein acetylation altered during viral infection? And subsequently, we wanted to begin to drill down on understanding the functions of some of these acetylations and whether they played a role in host defense or perhaps are actually important for facilitating viral replication. So we decided to focus on the prominent human pathogen, human cytomegalovirus, or HCMV, which infects over half of the adult human population. And while it's asymptomatic in most healthy individuals, it's a major health concern for people who are immunocompromised, and it's the leading cause of virally induced birth defects. Despite these factors, we have no effective treatments and no vaccine currently available. So this virus replicates over 4 days, which is quite a long replication cycle. And throughout its replication cycle, it interfaces with numerous cellular organelles. But I want to draw your attention today to the nucleus, where it has much of its viral replication cycle occurring, such as the replication of its genome and the packaging of that genome into viral capsids. So to really begin to address how protein acetylation was changing in time and space during HCMV infection, we employed a proteomic approach in which we infected primary human fibroblasts with HCMV and collected samples throughout the time course of infection. We then extracted the proteins, performed a trypsin digest, and then followed that with an anti-acetyl-lysine immunoaffinity purification and then mass spectrometry in order to determine in a site-specific manner how acetylation was changing on both host and viral proteins during infection. So today I'm going to be focusing specifically on some of our findings from the nucleus, where we observed here each line represents a single acetylation site, and we're progressing across infection time. And while we observed that many sites displayed decreases in acetylation during infection, we were very interested in this subset of sites that displayed increased acetylation, particularly at late time points of infection. And when we looked into these, we found that several of these sites were located on nuclear laminar proteins. So the lamins together form a meshwork at the inner nuclear periphery that serves multiple functions, including helping to stabilize the nuclear shape, and they also interface with host chromatin. We were especially interested in these proteins because during HCMV infection, it's known that the viral genome is packaged into capsids inside the nucleus, but those capsids are too large to fit through a nuclear pore, which means that the nuclear lamina has to be disrupted to allow for those capsids to effectively egress from the nucleus. And I will also comment here that there's really an emerging field of understanding post-translational modifications of lamins and how they might function in many different cellular contexts. But prior to our work, there was no non-functional role for acetylation on lamins. And so we started asking if some of the acetylation sites that we identified may have a function during infection. And today I'm going to be sharing with you our findings with regard to one site, K134 acetylation on lamin B1, where we observed increased acetylation throughout infection. And when we generated either acetyl mimics or charge mimics, what we found was that in the presence of the acetyl mimic, there was a significant reduction in the amount of virus produced compared to the wild type, leading us to hypothesize that this acetylation had an antiviral function. But we then wanted to understand mechanistically how this might be occurring. And so we employed microscopy to look at the morphology of the nucleus. And in the presence of wild type lamin B1, we observed the induction of infoldings and blebbings of the nucleus along with nuclear curvature. And we observed a similar phenotype in the presence of the charge mimic. However, in the presence of the acetyl mimic, we observed an amelioration of this phenotype and a loss of those infoldings and blebbings and a reduction in curvature, leading us to conclude that acetylation at lysine 134 on lamin B1 helps to stabilize the nuclear periphery. And subsequently, we've gone on to show that this is not unique to HCMV, but we're using herpes simplex virus type 1, we observed a similar phenotype of stabilizing the periphery, leading us to our model in which we propose that acetylation of lamin B1 impedes this disruption of the nuclear lamina during herpes virus infection, and thus impedes the ability of viral capsids to egress and acts as an antiviral mechanism. Now, while we were doing this work, we also noted that we detected acetylation of lamin B1 at this site in uninfected cells. And we found this really interesting, given that lamins have many functions. So we wanted to think more about if this acetylation could potentially have a role in homeostatic nuclear processes. And so I'm going to take a moment here to mention that lamins are really critical for nuclear health and for cellular health. And they've been implicated that when they're misregulated, they can lead to numerous disease phenotypes, including progeria, muscular dystrophy, and they've also been linked to cancer as well. And this isn't surprising when we think about the multiple functions that lamins can have, not just with regard to nuclear shape, but also they've been shown to be important for serving as chromatin organizing regulatory hubs by tethering chromatin to the nuclear periphery. And they've also been implicated in DNA repair. This was particularly interesting to us because recent work has shown that tethering of chromatin to the nuclear periphery is also important for certain DNA repair pathways. And if we think about DNA repair in cells, we know that effective double-strand break repair is really important for maintaining genomic integrity and preventing deleterious mutations. So given our knowledge of lamins, we hypothesized that lamin B1 acetylation may have an impact on DNA repair. And to initially test this question, we employed a COMET assay in which we have a head here of undamaged DNA, and then the damaged DNA will fan out as a tail. And the way we employed this was we treated these cells with bleomycin, which induces double-stranded breaks, and then looked at the persistence of breaks 3 hours post-treatment during that recovery period. And what we observed was in the presence of the acetyl mimic, there was increased persistence of DNA damage compared to the wild type or the charged mimic, leading us to conclude that this acetylation was leading to a delay in DNA repair. Now, this was also interesting because if we think about the link between DNA repair and the cell cycle, it's quite strong, given the fact that at the distinct cell cycle checkpoints, a key aspect of these checkpoints is assessing DNA integrity. So we next asked if this acetylation may also impact cell cycle progression. And by employing flow cytometry, what we observed was that there was an increased percentage of acetyl mimic cells in the G1 phase and a concomitant decrease in the percentage of cells in the other 3 cell cycle stages, leading us to conclude that this acetylation was leading to an activation of the G1S cell cycle checkpoint, and leading us to also propose that this acetylation may be impacting the assessment of DNA damage at this checkpoint. Now, something to keep in mind is that when DNA damage needs to be repaired, it often leads to the recruitment of additional proteins as repair factors. And so we hypothesized that this acetylation may be impacting the interactions between lamin B1 and other proteins, including DNA repair factors potentially. But one of the challenges we faced here was that because lamin B1 is so closely associated with the nuclear periphery, traditional immunoaffinity purification techniques are challenging because the stringency of lysis buffer that would be required in order to disrupt the lamin B1 nuclear periphery interaction would also effectively disrupt lamin B1's interactions with other proteins. And so instead, we employed an alternative approach using mini-turbo. What this allowed us to do was to fuse a promiscuous biotin ligase to lamin B1 as either the wild type, the acetyl mimic, or the charged mimic, and then add biotin, which then led to the biotinylation of proteins that were in close proximity to lamin B1, which we could then isolate with a streptavidin IP and use mass spectrometry to identify these proteins. And so here in this interaction network, the proteins that had increased association are shown in yellow, and those with the decreased association are shown in blue. As we progress across each node, we're looking at the comparisons between the acetyl mimic and the wild type, the acetyl mimic and the charge mimic, and the charge mimic and the wild type. There are two main clusters I want to draw your attention to. The first of which are the nuclear periphery proteins, where we observed an increased association between the acetyl mimic lamin B1 and these proteins, which is in line with our previous findings, where we showed that acetylation of lamin B1 helps to stabilize the nuclear periphery during herpes virus infection. Subsequently, we also had an interesting second class of proteins, which are these chromatin organizing proteins, where we observed that there was decreased association between the acetyl mimic and these proteins, suggesting that there's a dissociation between chromatin and the nuclear periphery in this case. And we've subsequently shown that this dissociation is independent of whether that chromatin is heterochromatin or euchromatin. This was especially interesting given the fact that, as I mentioned earlier, certain DNA repair pathways have been shown to be linked to tethering to the nuclear periphery. So we next wanted to know which DNA repair pathways may be impacted by this acetylation. And we thought about how at this G1S cell cycle transition, the predominant form of DNA repair is non-homologous end joining, which can be subdivided into either canonical or alternative end joining. And so we wanted to know if one or both of these pathways may be impacted by lamin B1 K134 acetylation. To do this, we employed reporter cell lines, the first of which allowed us to specifically look at canonical non-homologous end joining, as when the double-strand break is induced, if the break is repaired properly by canonical non-homologous end joining, GFP will be produced, and we can use flow cytometry to assess the percentage of GFP positive cells. And here what we saw was that there was a decreased percentage of GFP positive cells in the presence of the acetyl mimic, leading us to conclude that this acetylation is impeding canonical non-homologous end joining. We next wanted to look at if alternative end joining was impacted. And to do this, we employed a reporter that allows us to look at all end joining, so it encompasses both of these pathways. And here what we observed was that there was no significant difference among these cell lines, which led us to hypothesize that alternative end joining may be compensating for this impediment of canonical non-homologous end joining, which then led us to hypothesize that this acetylation may in fact be biasing toward alternative repair pathways. To directly test this, we again used these reporters, but because we know exactly where these double-strand breaks are being induced, what we could do was employ sequencing to then determine precisely how this DNA sequence is being repaired. So we could then determine if there was repair that was faithful, or if these sequences were being repaired by alternative methods that can be hypermutagenic because they may lead to a loss of DNA. What we ultimately observed was that in the presence of the acetyl mimic, there was no faithful repair, and over 90% of the breaks were repaired either through trimming or end resection, both of which are these alternative pathways that can be hypermutagenic. And this led us to conclude that this acetylation of lamin B1 is in fact biasing DNA repair away from canonical non-homologous end joining. Now, this really made us take a step back and ask, why might this be beneficial to a cell, given that these pathways can be hypermutagenic? And we thought about the cell cycle and when different DNA repair pathways are available. So for example, canonical non-homologous end joining is available throughout the cell cycle. However, homologous recombination, which is dependent on the replication of the genome, is only available in S and G2 phases. And so we hypothesized that lamin B1 acetylation may be changing in abundance during the cell cycle. Now, most of the cells are in G1 phase, and so in order to enrich for cells that were in S and G2 phases, we treated with a CDK1 inhibitor. We then employed targeted mass spectrometry to allow us to quantify the K134 lamin B1 endogenous acetylation in these different cell cycle stages. And what we observed was that there was nearly a threefold increase in lamin B1 K134 acetylation in the S and G2 enriched cells compared to the predominantly G1 phase cells. This leads us to conclude that this acetylation is increasing in S and G2 phases when homologous recombination is the preferred DNA repair pathway. And we then propose that this acetylation may in fact be serving as a toggle between different DNA repair pathways by mediating chromatin tethering to the nuclear periphery. We had one final question, which was mechanistically, how is lamin B1 acetylation biasing against canonical non-homologous end joining? Now we know that a key important factor in canonical non-homologous end joining is 53BP1. And so we looked at 53BP1 dynamics. And what we found was that upon induction of DNA damage, that all 3 cell lines displayed an induction of 53BP1 foci above the untreated baseline levels. However, as recovery occurred, while both the wild type and the charge mimic cells were returning back toward those baseline untreated levels, there was a persistence of 53BP1 foci in the presence of the acetyl mimic. Additionally, we observed distinct phenotypes for 53BP1 foci, and notably found that while there was a rapid transition from the diffuse and mixed phenotype to the clear distinct foci phenotype in both the wild type and the charge mimic cells, there was a delay in this transition in the presence of the acetyl mimic, leading us to conclude that 53 recruitment to sites of DNA damage and then ultimate resolution of these sites of DNA damage via this canonical non-homologous end joining pathway are both delayed in the presence of this acetylation. And so putting all of this together, what I've shown you today is that we performed an acetylome analysis that allowed us to determine how protein acetylation was changing in time and space throughout the time course of HCME infection. And from this large proteomic scale analysis, this led us to identify the first functional role for acetylation of lamin B1 in stabilizing the nuclear periphery during herpes virus infection. Moreover, by combining additional proteomic techniques along with some cell biology techniques, we've gone on to show that this acetylation of lamin B1 also has a functional role in homeostatic cells by both mediating chromatin tethering to the nuclear periphery and serving as a toggle between different DNA repair pathway choices dependent on cell cycle progression. And so this really shows the power of how we can integrate proteomic techniques to allow us to functionally characterize PTMs, and then this can lead to novel insights into both fundamental biology and how that might apply to disease states, as lamins are super important for many different diseases. And with that, I want to thank my advisor, Ileana Cristea, the rest of the Cristea lab, especially Josh, Jamie, and Pranav, who worked with me a lot on this project and our funding, and I'd be happy to take any questions.
Thanks very much, Laura. Very interesting talk. So I am now going to read out the questions for you. So the first one is, could you highlight what kind of quantification strategy was employed for acetylation time course study on HCMV infection? Well, is it LFQ, SILAC, DIA? Also, can you comment what type of normalization approach was used for quantification?
Yeah, so that initial acetylome experiment was a DDA experiment, so we didn't have any labeling. But in parallel with the analysis of acetylpeptides, we also performed an analysis of protein abundances. And this was mentioned, a similar technique was mentioned in an earlier talk with regard to phosphorylation, looking at both the PTM abundance and the protein abundance, because then we could normalize the changes in acetylation to those changes in protein abundances, which allowed us to then determine if the changes we were seeing in acetylation were due to just changes in protein abundance or if they were really happening at the PTM level.
Okay. So the second question is how many peptides input is needed for the acetylpeptides enrichment? Is the enrichment antibody from cell signaling company?
Yeah, so it was the cell signaling PTM scan kit that we used, which is a mixture of a couple different antibodies and has been used in several different studies and worked well in our hands. We had approximately a little over a 30% enrichment, which is really in line with other PTM studies for acetylation. And let's see, with regard to the input, so one of the current challenges with regard to acetylation is that because we really can only get about a 30% enrichment currently, which it'd be great to extend that to a higher enrichment, that you do have to start with a pretty big input. And so we were starting with approximately, at the beginning of our workflow, approximately 20 milligrams of protein.
And well, so we have one follow-up question. So how exactly have you normalized to the protein level with or without including standard deviation for both proteins and PTM peptides?
So what we were doing there was we had our acetyl peptide data set, and we had our proteome data set. And so for the proteome data set, I should say for both data sets, we initially normalized to the median abundance, which allowed us to normalize for any variability in the run itself. And then we also factored in that for the acetyl, we had to have a certain number of time points and replicates in which the peptide was detected. So this increased our confidence in that site. Of course, we were dealing with an infection time course. So we had 4 infected time points, and then we had an uninfected. And so once we had done that normalization to the abundance for that given run, then we normalized to our uninfected time point. So that was a first full joint over uninfected, and then we took the uninfected, or the time point of interest over uninfected for the acetyl, and then we divided that over that same comparison for the proteome.
Okay, so thanks so much again for sharing your work with us. I'll now pass this to our next chair, Dr. Fernando Corrales, for introducing his speakers.
Okay, thank you, Maggie. So my name is Fernando Corrales. I'm actually working at the National Center of Biotechnology here in Madrid, in Spain. I have to say that it's a great pleasure to be part of this fantastic adventure that is the Human Proteome Project. So I encourage you to take part of it. It's really fun, and you make friends. That is the most important thing. So I'm really glad to introduce our next speaker. He's Professor Andy Tao. He's professor at Purdue University. He's professor of biochemistry, chemical biology, and analytical chemistry. And he's doing an outstanding research on protein phosphorylation. Actually, he managed to link the technological side with the biological, biomedical discovery, the generation of knowledge, development of applications, and so on. So we have today a very good example of his job about phosphoproteomics and intracellular vesicles. He's asking if this could be the generation of protein biomarkers of disease diagnosis. So Andy, it's all yours. Go ahead, please. Thank you very much for being here.
Thank you. Thank you, Fernando, for the nice introduction. And I certainly would like to thank Alina for organizing this very nice webinar. Yes, so 2 previous speakers already talked about the phosphorylation, so I try to come from a different angle. So actually my title, as Fernando mentioned, that's going to be present a question to the panel and the audience as whether protein phosphorylation, phosphoprotein in extracellular vesicle represent the next generation of protein biomarkers for disease diagnosis. Well, so in order to talk about it for next generation, so we're going to talk about the current protein biomarker, the status of protein biomarkers, especially related to mass spec as a tool to identify them. And certainly we may introduce the protein phosphorylations in extracellular vesicles, work in my lab. And then also one major one is certainly talking about isolation for extracellular vesicles and for proteomic analysis. In the end, I may talk about recent work related to the neurodegeneration disease, particularly Parkinson's disease. Okay, so 2015, so this in clinical chemistry give a nice review on the application of mass spectrometry in clinical field. So as most of us are in the field using mass spec as a tool. So this is a particular for the application in clinical field. So you look over it, so most of them are actually using for small molecules. Very few for large molecule like proteins. So this is 15. So later on, 2018, there's one for ovarian cancer. So indeed came out through the work of large scale experiment. FDA approved for in vitro diagnosis multivalent basic panel for proteins and potential biomarkers. But this is a relatively new, and the actual current, let me see, hold on. Current status of really cancer biomarkers, I use the cancer as an example. So here, so indicating that, so in order to qualify as a, especially for in vitro diagnosis markers, that's he's showing the pictures. So usually instead of going for the direct solid tumors for needle biopsy, so you take the blood or other biofluids such as urine samples, try to identify markers for disease. So way is that all these markers. So I think I listed some of here. I think most of you guys know these, like PSA for prostate, CA125 for mainly for ovarian cancer, and other proteins. So these are considered the for protein biomarkers. But so here's a catchphrase. So most of these these markers have no screen value, so they can be used for basically for example follow-up for some of these chemotherapy, see whether or surgery whether being effective, but they don't typically cannot be used for screen purpose. PSA is considered most successful protein markers, but we know the reports say PSA is a lot of false positives, so still not very good marker. So really we have a long way to go to develop protein biomarkers for in vitro diagnosis. So why is this so difficult? So here's showing the sort of analogy of the proteins in our biofluids. Here's in the humans. So we all know that there are a lot of these proteins, but most of proteins are highly abundant proteins such as albumin or other proteins. So 99% protein are these top 20 proteins, and these actually related to disease markers. We believe they are actually much low abundant. It's buried in the deep in the ocean of the iceberg, and then typically if you analyze by mass spec, is typically overshadowed by these highly abundant serum plasma proteins. And so today I'll talk about try to develop a phosphoprotein as biomarker. This is even more difficult to see because they are really, really low abundant even. And then here's even worse is one of the enzyme alkaline phosphatase, which is typically released by our liver, is more highly abundant in our blood sample, so that they will hydrolyze all our phosphoproteins. So the point is very few phosphoproteins you can be identified in biofluids. So then here comes so certainly my lab's recent work, and I feel this is certainly exciting field. This is extra vesicles. So because of time and limits, I will not give a lot of introduction, but extra vesicles, this is certainly a contemporary hot field, ongoing field, a lot of research in both therapeutic biology and also in diagnosis. So what my lab has recognized that so extravascular certainly come from all different cell origins, and some of them certainly come from the cancers for other disease cells. And because of these double membrane, basically lipid structure, so we figure there's a possibility that these a lot of phosphoproteins actually being protected from the biofluids or these phosphatases in the biofluids, so that we'll be able to identify these phosphoproteins from these biofluids. Okay, so this is the work. Now it's more than 5 years ago. So these are the we reported the first large studies look at the in the actually blood plasma samples using breast cancer patient and also the healthy controls to look at the phosphoprotein, basic phosphoproteomics in extravasicles. And so we use the sort of standard high speed and ultra high speed centrifugation to isolate both exosome and microvesicles. These we all call extracellular vesicles, and then go through the standard isolation of phosphopeptides, and then in the end to do label-free quantitation. So the procedure is relatively straightforward. Since it was published quite a few years ago, so on the report, so this is the first is that a lot of phosphorylations in these samples. Actually, when we first get the data, I couldn't believe it. So in about 1 or 1/2 ml of plasma samples, we can identify over 3,000 phosphopeptides at that time in the Fellows Pro instrument. And then we all combine multiple samples, we identify close to 10,000 unique phosphopeptides, represent over 2,000 phosphoproteins. And among them, certainly we identify over close to 200 phosphoproteins are disease-specific. And we compare these disease-specific phosphoproteins against these certainly known. So there are quite a few studies, mainly previously based on these tissue samples, cell culture on breast cancers. We found that actually over 2/3 of these phosphoproteins have been reported related to onset and progression of breast cancers. So certainly that we demonstrate the feasibility of using extravesicals just from blood sample. You can identify these phosphoproteins, and we also look at these simple pathways. We identify actually a lot of the signaling pathways related to cancer onset progression. So this I consider quite significant, because for a lot of biomarker discoveries these days in biofluids, usually these proteins are statistically different, but a lot of meanings are usually related to the immune response. That's pretty much it. But here we identify certainly a lot of them are actually being previously established, these pathways related to the cancer onset progression. Okay, so since then we have a set of studies, so recent studies we published in Nature Protocol last year, considers PTM webinar. So I present a protocol which allows to use a single clinical samples, since these days why we use most of samples from clinical and patient samples, so that sometimes these samples quite precious. So we try to develop an actual method. You can look at multiple PTMs in a single sample. So here's basically you can sequentially isolate phosphoproteins, glycoprotein, and acid proteins, or potential other modified proteins for single source of clinical samples. And so the data actually compared to if you do in parallel, more or less still similar. So which means that you really can save these a lot of clinical samples. You look at multiple PTM studies. First, you save this clinical sample. Number one. Second, you potentially can integrate different PTM studies, look at the diseases. So, so all these studies certainly we present is based on the sort of standard called centrifugations and ultra high speed centrifugations. So these are certainly, it's good for screening in the lab, but probably not very good for clinical studies. So we spend a bit of time for past 3 or 4 years to look at the different isolation approaches, especially develop these automatic method or high throughput approaches for extracellular vesicle isolation. But 3 years ago, so we came up with a chemical affinity approach to isolate basically extracellular vesicles and then for follow-up all the phosphoproteomics and also proteomics analysis on extravesicles. So this is the study on the urine samples. So we started with urine, actually a little bit easy to deal with because there are a few of these plasma protein or urine proteins inside of which can potentially interfere with our mass spec analysis. So since it's magnetic beads-based, so things that make it pretty easy. So you throw the magnetic beads into the urine sample and incubate for much short time, and then just isolate magnetic beads then for ongoing analysis. And then it took us for another 2 years to basically come up with a workable protocol using the magnetic beads for the urine, not urine, for the plasma and the serum samples. And as I mentioned, they are a little bit more complicated, but we finally work out the method that can be applied to the urine for the plasma sample as well. So here's some data. Basically, for extravesical field, you need to demonstrate that you can really isolate the extravesicals by basically measure these markers. So here's showing some CD9 markers. So here's an example showing that for if you use standard centrifugation, you get about 20% or less isolation efficiency. But if we are using a better method, this is magnetic beads based, where you can get over 90% of efficiency, and based on the CD9 or other markers for extravasicals. So here's showing that for other markers, so you can really see the specificity of the isolation. And finally, the data indicating that if we use the magnetic beads-based method compared to the centrifugation method, you can really improve the sensitivity, and coupled with our polymer-based phosphopeptide enrichment, we can get over, some of the cases you can get over 40% of signal enhancement. So this is certainly significant for clinical sample applications, that you can get a good sensitivity for these low abundant phosphoproteins from extravasicals. Okay, so we sort of come up with another generation magnetic beads based, so which is published last year. So this is a little bit different. So this is based on the idea that using two function groups on the magnetic beads. One is titanium. So the reason we use titanium can more or less kill it with phosphate group in the phospholipid. Number one. Second one, we also introduce a basically phospholipid mimic reagent called D-HPP, and that can insert into the membrane of the EVs, so that using basically two function groups in order to better isolate the extravasicals. So after that, so the following method will more or less the same. So here's showing the example. Actually, it works nicely for us, especially compared to the singly functionalized. Here's the name may be a little bit strange. So here's indicating the for the D-HPP only magnetic beads, then versus titanium based magnetic beads. If we have both function groups, you can really increase the isolation by, we look at the Western blotting using CD9, and here's showing the total protein. You can isolate almost a double amount of peptides isolation efficiency, and compared to the ultracentrifugation, you can get almost more than triple amount of phosphopeptide identifications in the samples. So here's showing some of the identification for the top 100 and also all the other exome proteins, so indicating that we'll be able to isolate these samples efficiently using magnetic beads based isolation. Okay. And for final applications, I'm going to talk about try to identify these proteins by fluids for Parkinson's disease samples. This is actually not original initiative from our group. We published a paper in 2018 in the urine samples. Basically, we identified these phosphoproteins, and then we didn't actually look deeper into the samples which are published in JPI. Then Michael J. Fox Foundation contacted us. The reason is they found we identify in the urine sample the LAC2 phosphorated LAC2 phosphopeptides in the samples. So they feel that certainly our method seems like quite sensitive, and so they would like us to work with them and ask us whether we can work with them. Certainly yes. And so that's a slow collaboration with Michael J. Fox Foundation, who provides the money, and the Columbia University provides the sample. They have called a LAC2 consortium, which includes both all these samples have 4 groups, so which is a healthy control, and then with LAC2 mutation, but without Parkinson's disease, and then LAC2 without mutation, but with a disease. In the end, is LAC2 mutation with disease. And all these 4 groups, and so tutors 80 samples, they send it to us, actually randomly sent to us. We basically analyze one by one. And so the procedure is, in this case, we isolate by the magnetic beads, the EV trap, and then followed by, as I mentioned, standard more or less protocol for isolation peptides for mass spec label-free quantitation. So LAC2 is, we know this previous speaker and Claire mentioned that pink one is one for them, LAC2 is another one. So actually LAC2 mutation represents about the most abundant considered mutations in Parkinson's disease, but most abundant is only still 1 or 2% of the mutations. So this is actually not a strong genetic basis, let's say. And that's why I also presented today that the PTM studies is very important beyond the genomic studies. All right, so here's, because the time limits, I give a very quick summarize of data. So we identify quite a lot of certain samples with phosphopeptides in terms of tutor identification. So over 80 samples, we identify over 10,000 phosphopeptides and also over 2,500 phosphoproteins. And then since we have 4 groups, we can basically, after the analysis, we can group them into different clusters depending on the distribution among these 4 groups. For example, disease markers indicating that they are either without mutation or with mutation, but all have the diseases, Parkinson's disease, and then the genotyping basically in mutations, but not necessarily with the disease. So we can come up with different markers considered as disease marker or risk biomarkers. So that's certainly quite interesting for us to look at that. And then we certainly eventually, through this study, so we identify a panel of these biomarkers, and these biomarkers we give them certainly look at the ROC curves. If we have 3 or 4 added together, actually give a quite good selectivity over ROC value of over 80% or some of over 90% consider the overall value specificity and sensitivities are pretty good. And in the end, so we also compared with some of Parkinson's disease clinical parameters. So here's indicating one of the specific look at the overall the Parkinson's disease multi-parameter together. These are ones so look at the correlation, the Pearson correlation. So here's some of these proteins we identify markers that have good correlations with the clinical parameters. So here's indicating some of these proteins that actually in our data basically showing that a good correlation with the clinical parameters. In the end, so we have using artificial intelligence modeling as look at the first set of data as training for control versus disease, and we have 24 healthy and 24 Parkinson's disease, and then use the panels we developed so we can get 100% accuracy for the training set, then use that for testing set. So there's about 25 of these, 23 of tutor we get over 95, 96. Close to 96% accuracy. Basically, one of them we didn't predict as the disease, but the rest of them will all get a correct prediction of the disease. So indicating that certainly the phosphoproteins are, there is great potential to develop as a potential biomarkers for the diseases. Okay, so I finished my talk here. I would like to thank certainly for the past multiple years, and we are digging into this extravascular field to develop phosphoproteins as potential biomarkers. So we certainly collaborate with multiple groups in different clinical field, so can center funding, including Michael J. Fox Foundation for the recent studies. Thank you.
So thank you very much, Andy, for this outstanding presentation. Let me just read the questions we have for you here. So the first one is, have you observed whether sequential enrichment for 2 different PTMs causes bias in the second enrichment PTM compared to the same PTM being enriched first?
Yes, so we have in the nature protocol, we had a comparison. So certainly a lot of PTMs are antibody-based. So antibody-based is a relative, you don't introduce contamination, just incubate with different ways. So usually I think it's quite compatible as long as you don't get a non-specific bonding of them, then certainly it's fine. And for the phosphor versus the glyco, I believe we first did the phosphor enrichment followed by glyco enrichment, because glyco, we didn't use the affinity at that time, we used the basically hydrogen chemistry to do that, so which is a bit of harsh, so we do that in the second step. But overall, I believe we can do sequential enrichment based on our analysis, more or less in line with if we do it in parallel. So that's providing a good advantage that you can use a single plasma sample, you can do multiple PTMs.
Mm-hmm. So I have a second question for you. That is, how much urine input per sample?
Yes, so that's a good question. So it took us a really while to figure out. This is certainly still ongoing development. This is also the challenge for extravesical based field. The first is, so once we get extravesical, I believe it is relatively stable, but during the collection of the sample, a plasma sample, and storing the sample during that period of time, I think we found that there are a lot of variabilities. So how long you live in the room temperature, and when you get to store them, and especially in the clinical conditions, sometimes they don't store them immediately, and basically centrifuging down. We work at one time with medical school. They want to be very careful. When they collect the blood, they immediately put 4 degrees, but actually made things much worse. And so actually you don't want to store them in 4 degrees because the extravesical can precipitate out with something else. So it took us for a while to figure out. But we analyze, get the biomarker, basically let's use plasma sample as an example. We get from different biobanks. Actually, majority biobanks sample are quite consistent, which I'm glad to know that. So for phosphoproteomics, and so we need really quite a bit of more. We typically need about 1 ml of plasma to get quite a decent phosphoproteomics. But if we only do proteomics sample, I think you only need about a few hundred microliter is good enough of plasma sample. You get a very nice data. But phosphoproteomics, certainly you need quite a more samples to do that. But I think this is called discovery phase. Once we move into the called targeted, more like validation stages, when you do targeted, you can have much better sensitivity. You don't need as much sample.
Let me just ask you the last one that might be related with this one. Is how do you normalize exosome samples since there is a lot of variability? Yes,
that's actually a good question. When we jump into this field about 5 years ago, that was really a lot of groups just, especially microfluidics, they just by measuring the extravesical as biomarkers. And then we found that if we collect the extravesical from same patient at a different time point or different days, you have variations. So in that case, by measuring extravesical only, none of them we don't think this is a very reliable one. So in the end, what we do is, especially for phosphoproteomics method, so we normalize the protein to start with. So after that, we basically measure the phosphorylation difference. So that gives us a little bit better reproducible data.
Okay, thank you very much, Andy, for your talk and the discussion. And I think we shall move on to the next chair. So Frank.
Yeah, thank you so much. So my name is Frank, and I'm working at the Y Cornell Medicine in Qatar. And I just like to, it's a pleasure for me to introduce Stacy Malaker from the University of Yale. So Stacy is working on the development of mass spectrometry tools and even analysis tools for the mucin domain glycoproteins, and the talk is revealing the cancer-associated mucinome. So we are looking very much forward to your talk, Stacy.
Thank you so much for the introduction, and thank you for having me here today. I just started my lab at Yale about 3 months ago in January. So I'm actually going to talk today about a lot of my postdoc work, which was performed in the lab of Carolyn Bertozzi at Stanford. So Nicki did a great job introducing all of us to glycosylation and different types of glycosylation earlier today. So instead of giving you that, background, I'm just going to jump right into my favorite type of protein, which are mucins. So mucins are characterized by having a high proportion of prolines, threonines, and serines in their so-called PTS domains. And I use PTS domains and mucin domains kind of interchangeably, so just know that that's what I'm referring to. Within these domains, there's extremely dense O-glycosylation. So most of the serines and threonines are in fact modified. And this contributes to this extended and rigid bottle brush-like structure that can either extend away from the cell membrane or form these gel-like secretions on the outside of the cell. Now, there's a family of canonical mucins. So mucin 1 is MUC1, mucin 2 is MUC2, et cetera, for a total of 20 canonical family members. And we know that these canonical family members are very, very intimately tied to disease. So to be sure, over 90% of breast carcinomas upregulate MUC1, and MUC16, also known as CA125, is one of the only available biomarkers for ovarian cancer. And now we know in mouse models that these altered mucins are somehow linked to tumor genesis and immune evasion and chemotherapy resistance and so on. But despite the fact that we know this at sort of a global level, right? We know that the mucins are upregulated, or we know that the glycans change in some way. We don't actually know how the tumor microenvironment modulates the sites and the site specificity and the glycan structures on these proteins, because mucins are incredibly difficult to study, especially by mass spectrometry. One of the main challenges in studying mucins by mass spec is that, our workhorse normal proteases like trypsin or gluci or aspen or what have you can't actually access the backbone due to the dense glycosylation. And so they won't actually digest these densely glycosylated mucin domains. So when I joined Carolyn's lab as a postdoc, we hypothesized that due to the ubiquitous nature of mucins, that there had to be an enzyme or a class of enzymes that were actually able to come in and cleave these into peptides that are more amenable to mass spec analysis. So enter sticky. Now, sticky is secreted by E. coli in the gut to chew up gut mucin so that the E. coli can actually get into the cells and cause virulence. So it was reported in the literature that sticky is able to digest mucin domain glycoproteins, but doesn't work on non-glycosylated or sparsely glycosylated proteins. So when we first got our hands on it, we wanted to show that that was in fact the case. And so we took 6 recombinant mucin domain glycoproteins shown along the top here. And you'll see that every time that sticky is added, we get a pretty significant digestion event occurring. However, when you add sticky to BSA, which is not glycosylated, we don't see cleavage. And if you add sticky to fetuin, which is glycosylated but not a mucin, we also don't see cleavage. So sticky is in fact a mucinase, a protease that is selective for mucin domain glycoproteins. So once we had essentially shown what was already in the literature, I wanted to figure out how exactly sticky is cleaving. And there we go. Okay, so what I found is that sticky has a distinct peptide consensus motif for cleavage. So it will cleave N-terminally to serine or threonine at this P1 prime position if there is a serine or threonine 2 residues away from that at that P2 position. However, the peptide sequence wasn't actually sufficient for cleavage. That P2 position was invariably glycosylated. So that had to be glycosylated. And we detected a number of different glycoforms on that P2 position, ranging from a single GalNAc all the way up to this disialylated structure. We also had other data that suggests that it also cleaves peptides that have large core 2 sialylated structures. Importantly, too, pretty much any position along the peptide backbone could be glycosylated, but only that P2 position actually had to be glycosylated. So once we had solved the consensus motif, and we know that that consensus motif is likely to reside in pretty much every mucin domain, I wanted to show that it would be useful for mass spec analysis. So I took those same 6 recombinant mucins that I just showed under 4 different enzymatic conditions, and I manually sequenced many spectra. And by comparing the peptides present in the sticky versus non-sticky treated samples, I showed that we can increase our sequence coverage of the proteins by about 15 to 20%. But we really impressively saw a difference in terms of the number of glycosites that we could actually assign. So that went up by over 250%. And then even more impressively was the number of localized glycans to those various sites, which we increased by over 300%. So sticky is very useful for mass spec analysis. Now, all of this was actually published in 2019, And in the meantime, we've been working on 2 follow-up projects, if you will. One is expanding the mucinase toolkit and using these mucinases as visualization tools. And the other one, which I'm very excited to talk about, is the enrichment of mucin domain glycoproteins from complex samples. But I'll just quickly go through some of our new mucinases, which are shown here on this very busy slide. But all of this to say that we now have 7 characterized mucin domain glycoproteins, and they all, interestingly, are reliant not only on the peptide sequence, but also the presence of glycosylation. And perhaps more interestingly, all of them are just slightly different. AMO627 will cleave in between 2 glycosylated residues. ZIMP-C will cleave 4 residues away from the glycosylated residue, and so on and so forth. But due to a number of different reasons, sticky is our favorite mucinase because it's easy to express, and it's what we call a pan-mucinase because it's relatively agnostic to the type of glycosylation that's present, and it seems to work on pretty much every mucin that we've worked with. We also work with this BT4244 because it has selectivity for these truncated glycans that are known to be present more often in cancers. So the graduate student I was working with, Judy, decided to turn sticky, or a point mutant of sticky, which is capable of binding mucins without cleaving them, into Western blot probes. So if you see here a total protein where we have BSA, which again is a non-mucin, fetuin, which is a non-mucin, and then 5 mucin domain glycoproteins, what you'll see in the total protein is that we can pretty much see everything except podocalyxin. Now, when you use sticky's point mutant to blot, what you'll see is that we don't see these non-mucins anymore, and we do in fact see podocalyxin as well as all of the other mucins. So we're using these as sort of pan-mucin blot probes. Even cooler, in my opinion at least, is that we're using these mucinases for immunohistochemistry. So the current method of staining mucins is this non-specific AB pass staining, which essentially just stains anything that's negatively charged. And so you get all of this purple stain, which is pretty non-specific, and you can even get some cytoplasmic staining. And so this is in the gut, and it's known in the gut that the primary mucin producing cells are these goblet cells, which are shown here, and then the secreted MUC2. And you can see that our stain is much more specific for the actual mucins versus anything that's negatively charged. So that was also published earlier last year. And the last thing I want to talk about today is defining the mucinome and enriching mucin domain glycoproteins from complex samples. So I mentioned earlier in the talk that there is a canonical family of mucins, MUC1, MUC2, MUC16, so on and so forth. However, there's a whole slew of other proteins that are known to contain a mucin domain, such as PSGL1 or CD44 or CD43, but a total list of the proteins that contain a mucin domain has never existed before. And so when we started this project, we reasoned that because sticky can, or the point mutant of sticky can bind mucins without cleaving them, we could use this as an enrichment tool to go through and define the mucinome. So sure enough, we took the inactive point mutant of sticky, conjugated it to beads. You can add your sample, complex sample like cell lysate, bind the mucins, and elute off and analyze by Western blot or mass spectrometry. So in analyzing by mass spec, well, before I go any further, I'll just say that these volcano plots are always usually pretty busy to me. So anything to the right of this vertical line is over 5-fold enriched in the elution of the enrichment. And anything above this horizontal line has a p-value of less than 0.05. So anything in this region here, we consider statistically significantly enriched in the elution. So in just looking at these proteins here, we could go through and assign the canonical mucins, MUC13, MUC16, MUC1, as well as some other ones that are known to have mucin domains. But then the issue became, well, how do we know that all of these other proteins have or don't have a mucin domain? And so for this, we actually developed what we call our mucin candidacy algorithm. And this was spearheaded by my colleague, Nick. And so essentially here what we do is we take the whole human proteome and we put it into an O-glycosite prediction tool called NetOglyc. And this assigns what sites it thinks will be glycosylated. And then we take those sites and we remove anything that's previously known to be phosphorylated, because there's not known to be a significant overlap between GalNAc and phosphosites. We also take into account the cellular component because given that mucins are extracellular, transmembrane, or secreted, we don't really want to include anything that's known to be specifically intracellular. And then we go through our mucin candidacy algorithm, and I won't go into this a ton. I will just say that we preferentially bias our algorithm towards threonines over serines because threonines are known to be more often modified by GalNAc. We use the subcellular localization. And we also calculate a density because, as I mentioned on the first slide, mucin domains are known to have extremely dense O-glycosylation. With that, we then assign a mucin score and we can assign confidence levels to the various proteins that come out. In doing so, we estimate that about 2% of the human proteome actually contains a mucin domain, which is much more common than previously thought and is actually about the same percentage as the proteases in our proteome. So that's pretty cool. When you take the same data set that I just showed you and you put it through our mucin candidacy algorithm, we can now go through and label all of the high confidence mucin domains. What's interesting is that the high confidence mucin domains are preferentially enriched in the elution versus some of the low confidence mucin domain proteins, which are essentially left in the lysate. But once we saw that this was working, we were sort of off to the races. And I ended up enriching mucins from a whole bunch of commonly used cell lysates. And in all of these cases, you can see that the high confidence mucins are definitely found enriched in the elution. But then I just wanted to compare what we found in the different cell lysates. And this is effectively a very complicated Venn diagram, but I just want to point out that, if there's 1 dot, then these 6 proteins are found in this cell line. If there's 2 dots, then these 4 proteins are found in these 2 cell lines. But if there's 5 dots, then these 6 proteins were found in all 5 cell lines. So these 6 proteins are shown up here. And 4 out of 6 of those proteins were previously known to have a mucin domain. We have the canonical MUC1, CD55, et cetera. But 2 of the proteins were previously unknown to have a mucin domain. So what's cool about this is that our platform can be used not only to enrich known mucin domain proteins, but also discover potentially new mucin domain glycoproteins. So all of this, again, was done in cell lines. And in order to make this sort of more clinically relevant, we decided to enrich crude ovarian cancer patient ascites fluid. And so we used 5 patient samples. And you'll see 2 things. One is that the enrichment worked even better in these ascites samples in that there are very few, if any, mucins that were not enriched. And then additionally, as I'd mentioned earlier, MUC16, also known as CA125, is one of the only available biomarkers for ovarian cancer. And we found it enriched in pretty much, except that one, in pretty much every sample. And then perhaps most interestingly about these samples was that this is another fancy Venn diagram. When you compare all of these, there were 20 mucin domain glycoproteins that were found in all 5 samples, which is really interesting because what I didn't say about MUC16 is that even though it's the only known biomarker for ovarian cancer, it's not necessarily the best biomarker. And so we're hypothesizing that if we expand this patient cohort, we'll be able to define the ovarian cancer mucinome in order to develop better diagnostics or prognostic indicators. And then the last thing I want to go through really quickly is that in our ascites samples enrichments, we noticed that essentially this technique acts as a de facto O-glycoprotein or O-glycopeptide enrichment tool. So if you just look at the glycoprotein identifications in the ascites samples alone, it's 97% non-mucins, versus in the enriched samples, that goes up to 60% mucins. So it's in the majority. And then perhaps most interestingly is that if you just look at the glycopeptide PSMs, in the ascites samples, you have over 80% N-glycopeptide identifications, versus in the enrichment, you have over 80% O-glycopeptide identifications. This is just the same thing shown in a different manner. And again, we were able to identify a few new mucin domain glycoproteins. And even beyond that, we were able to identify a bunch of different O-glycopeptides that reside within our assigned mucin domain, which corroborates our thought that our mucin candidacy algorithm is in fact working. And finally, we were able to identify about 60 glycopeptides from MUC16, which could be used again in terms of diagnostic indicators. So with that, I'll just summarize and say that a point mutant of sticky can enrich mucins from complex samples like cell lysates and ascites fluid. This is accompanied by our mucin candidacy algorithm, which helps assign confidence to our identifications. And it works as a de facto enrichment method and has allowed us to identify hundreds of different O-glycopeptides in the elution. So I will just thank my previous laboratory, especially Nick, Kayvon, Judy, and our fearless leader, Carolyn, as well as my own small but mighty laboratory with my one graduate student, Valentina. And with that, I encourage you to follow our updates, and I'm happy to answer any questions.
Thank you so much, Stacy, for the very nice talk. So we have one question from Q&A. So do STAC-E result in long peptide backbones? If so, do you see a secondary protease to chop down the peptide backbone?
Yes, so sometimes that does happen. So normally I will follow a sticky digestion with trypsin as well, just to shorten it. And also that helps with, adding charge or making sure that there's charge in the peptides. I'm also currently working on sort of doing combinatorial digest with different mucinases with different specificities in order to sort of optimize the size for mass spec.
Yeah. So then I have another question here. Have you looked into the differences in glycosylation between cleaved in the plasma supernatant and uncleaved mucins?
I'm unclear as to what kind of samples they're talking about.
Yeah. Have you looked into the difference in glycosylation between cleaved and uncleaved mucins?
So in the enrichment samples, we're not cleaving because it's the point mutant of sticky. And so we're pulling down the entire protein. So they shouldn't be cleaved in the supernatant or the plasma.
Okay.
So I'm not sure. I hope I can answer that person's question, but I'm happy to do so offline.
Okay. So I have just a final question from my end. So you manual evaluated 1000 spectra. This would be a perfect training set. Did you try to use your manual evaluation for the algorithm to identify these peptides?
No, but the OPAIR program that my colleague Nick developed took those manual assignments into account because we had this whole database of things that I'd already manually assigned. So that was used for the OPAIR program.
Okay, excellent. Good. Thank you so much, Stacy. And with this, I would like to hand over to Ilyana for the next speaker.
Thank you, Frank. So it gives me great pleasure to introduce our last speaker, Professor Alexey Nesvizhskii from the University of Michigan. I have known Alexey for a number of years, actually probably for over 13 years. Alexey and I have co-taught courses on protein interactions either at Cold Spring Harbor Laboratory or at US HipA. And I feel that everyone actually knows Alexey in the proteomics field. He is a leader in the development of computational platforms for analyzing different types of proteomic data sets, SAINT, CRAPOME, MapDIA, PD nodes like MS Fraggers and Philosopher, TMT integrator, just to name a few of the computational tools that Alexey's team has developed. Today, he will tell us about another one, FragPipe, that is directly relevant for identifying PTMs. Alexey.
Well, thank you so much, Ilyana. I'm sharing my slides now. Hopefully, you could see them. And let me go to the slide presentation mode. So I'll talk about the FragPipe computational platform, but specifically with a focus on post-translational modification analysis. And before I get into that, I would like to sort of advertise the platform itself. even though we started with a rather limited application for this tool, which is to enable graphical user interface for a Nesvizhskii search engine, over the years, we actually developed it in a full computational platform where you can do pretty much everything from conventional database searching to label frequentification analysis with match between runs to TMT analysis to building libraries for DIA analysis and so on and so forth. So I really encourage you to try that if you haven't done so. Now, coming back to the post-translational modification analysis, to give a little bit of a background, my first project in this field 20 years ago when I started as a postdoc in the Rudi Abrasov lab was to look at high quality unassigned spectra and try to figure out what they were. And I was fascinated by this subject for many, many years now. And sometimes we refer to that as the dark matter of mass spectrometry-based proteomics. And this is in part what motivated us to develop now our new search engine called Nesvizhskii, as well as the whole pipeline for comprehensive PTM analysis to go after those spectra and figure out what they are. So Nesvizhskii, to me at least, is really this dark matter telescope for dark matter proteomics. And the FragPipe is the pipeline that fully enables this type of comparisons and analysis for pretty much everyone with very minimal computational experience. Now, the Nesvizhskii search engine itself was published 4 years ago now. And the key computational advance there was fragment ion indexing. It's not only specific to database searching and peptide identification part. In fact, this is how we speed up label frequentification, and it's because we can index the data for tracing peaks. So it's really the indexing part is a key for pretty much everything that we're doing these days. But of course, the main advance was that we were able to speed up searches by a factor of 150 times faster than conventional tools. And that enabled us to identify modified peptides through a different strategy than was sort of practiced before. Now, there are a lot of strategies for identifying post-translationally modified peptides. And in my 20 years in proteomics, I certainly tried all of them, from iterative multipath searches to sequence tagging followed with blind PTM searching to a so-called dependent peptide approach. But what I really like about open massive set database search strategy that I'll talk about in my presentation, and it's really because it's really an extension of a conventional search that we're all familiar with. And that's why when I think about our pipeline and Nesvizhskii as a search tool, I think about it as a Swiss knife tool for proteomics, because we really we're not developing something that's complicated or hard to understand. We hope to develop a tool that's applicable to all sorts of searches and scenarios. And I'm really glad that it works for PTM searches through this open mass of set search strategy really well. Now, what is this mass of set open search strategy? So let me explain this using this slide here, where if you look on the left, we see a conventional closed database search, where we basically search for peptides in a database that have a calculated MOEZ matching observed mass to charge ratio for a precursor peptide, right? Plus maybe a few unmodified peptides and maybe a few peptides specified as variable modifications, few modifications specified as variable modifications. Now in the middle is open search, is completely unconstrained strategy where we can search for any mass shift on a peptide, maybe from -100 Dalton to 100 Dalton, or from -200 to 1000, whatever you specify. And any peptide in a database will essentially be considered as a candidate for a particular spectrum. So we can identify any post-translational modification with any mass shift on the peptide. Now, massive set search is something in between. This is where we can pass a string of numbers and say, go and search, for example, for unmodified peptides, like in a closed search, but also look for any peptide that could be shifted by, say, 42 Dalton or 79, 96.96 Dalton, allowing essentially for acetylation and phosphorylation on any amino acid. So that's the massive search strategy, which is really great when we don't want to do a full search, but we want to really look for a very extended set of modifications. And in fact, we really love this massive search strategy for glycoproteomics as well as labile PTM analysis. Now, as we developed this tool that enabled open search strategy, we started exploring what kind of applications we can do with that. And of course, we've got a ton of collaborators and a ton of users of MS-Fragger in our pipeline that did things that we didn't even think about initially. But sort of this, as a quick summary of what you can do is that in a more straightforward way is that it's an excellent strategy just for a sample QC, for example, where you look at all mass shifts that you identify in your peptides, and you realize that you have some side chain labeling, some artifacts in the data that maybe you can go and revise your conventional close search to enable those additional modifications as variable modifications, for example. It's something you would do for a PE sample to figure out what additional modifications you would want to specify in your close search. But you can really extend it to biological applications where you look for protein RNA crosslinks. We have collaborations, numerous collaborations now in chemical proteomics domain, where we look at the site specificity of certain tags, where they bind, but also what mass shifts actually we have in peptides, and so on and so forth. So even for the most straightforward open search, there are already a ton of applications that one can explore and apply in practice. Now, I like this illustration of what we can actually find in open searching and how many of those peptides we have additional identifications that we get from this data when we go beyond conventional search using data generated on affinity purification mass spectrometry experiments, because in these data sets we enriched for proteins such as bait proteins. They're highly enriched, and we can identify a lot of post-translational modifications in those proteins. So here we reanalyzed data from the first Bioplex manuscript from Steve Gigi's lab. There's about 1,000 bait proteins analyzed using APMS, 64 million spectra. It would only take 3 or 4 days with a MS-Fragger to search this in an open search. If you take sequence to some other conventional tool, it would probably take you months, if not years, to open search like we can do with our MS-Fragger. Now, so it's really astonishing what you can see in data sets like this. So what I'm plotting here is a scatter plot where on x-axis I'm plotting number of PSMs, peptides we see, PSMs, peptide spectrum matches we see for bait proteins in a regular search versus how many PSMs we see if we do open search. And if you look carefully, you'll see that on average we get 4 times as many peptide spectrum matches for those bait proteins. For some, we have 50 times as many. So here I'm showing one example of ETH-E1 protein, where we go from 48 peptide ions in a conventional closed search to almost 2,500 proteins in open search, right? It's a huge boost in the number of IDs. And interestingly, if you take those proteins, bait proteins, where we see the most increase in the number of identified peptides in open search, so they're the most sort of post-translationally modified, and do gene ontology analysis. So in this data set, this gene ontology category, small molecule metabolic processes, came up as the most enriched. So those are probably, including this protein, is a part of this biological process. So we think that those proteins are modifying other proteins, and they modify themselves, and that's why the most decorated with post-translational modifications, and that's why there is the greatest boost in the number of peptides we identify for them when we open up the search space to look for all those mass shifts that we can find. So in the last few years, we actually worked really hard on improving the infrastructure behind all these algorithms. So we just published localization-aware open search that addressed the key limitation of open search strategy, in that we, in a conventional open search strategy, we do not include fragments containing modifications in scoring. We addressed that through localization of our open search algorithm published in Nature Communications last year. As part of that, we introduced MS1 and MS2 based mass calibration and search parameter optimization procedure. In a separate publication, we introduced ultra-fast de-isotoping algorithm that's really great for glyco data, and a few other applications. We extended it to FAMES as well as timsTOF data in a series of publications. We also introduced monoisotope correction. So it's really a work in progress where, you know, my team is working really a lot on improving all steps in the process. We also realized that we need to, you know, there is so much interest in doing these deep dives into post-translational modifications on a large scale. So we developed a computational tool called PTM-Shepherd for analyzing and summarizing post-translational and chemical modifications defined from open search. That's another publication that we just published in Molecular and Cellular Proteomics. Using this tool, we can identify for a particular mass shift, we can see where it localizes mostly on what sites in peptides. Also look at spectral similarity, retention time shifts introduced by the modification. See what's the abundance of modified peptide in comparison to unmodified counterparts. And this is really critical if you want to look for modifications, especially try to find novel or unexpected modifications in this data. So we, for example, one illustration on this paper, PTM-Shepherd paper that we just published, is when we took the data from Bernard Kuster's synthetic peptide study. And here we identified, among other things, we identify that there is a loss of 18 on lysine, where we did not expect it initially, because, you know, we can expect that loss of 18 as a neutral loss of water on any amino acid. And in fact, we do see neutral losses of water in general. And we see that for when they're localized to other amino acids or E, there's sort of, there is no retention time shift. Now, for pyro-E, when they're localized to N-terminal E, we do see that there is a population of peptides with a shifted retention time shift. So those are known modifications, this pyro-E modification. But what we observed the most is the minus 18 on lysine, C-terminal lysine. And in all cases, when we observed such a modification, there was a very significant retention time shift, suggesting that this is not a loss of water, but it's a real modification that happens in the sample. So digging deeper, we think that it's a sort of unappreciated artifact of peptide synthesis, where there is a cyclization event happens on a C-terminal lysine, because that's where their peptides is desynthesized, they're attached to the solid support. And before they cleaved, there is this cyclization event that happens. Now, might not be biologically interesting modification, but I think things like this are extremely important if you want to use synthetic peptides and MRM, PRM analysis as a way to do more accurate quantification and in general, better understanding things that happen in the samples. So we run it as part of the CPDAC consortium. For example, we run it on TMT data on hundreds of samples generated in different sites. And we use PTM-Shepherd and open searching to visualize differences in post-translational modifications between different data sets, different sites, between different cohorts, cancer cohorts. And we can identify, of course, again, sample preparation artifacts, where you could see how you know, a particular site, say the Broad Institute or Johns Hopkins Institute, the post-translational modifications that we see are, you know, specific to that site, probably due to, even though the sample preparation protocols are harmonized, each site still introduces some of the artifacts specific to that site. So we can see that. But in addition to the site-specific chemical artifacts, we do see biological modifications that are correlated between different data sets, such as phosphorylation and glycosylation and some other ones. So it's not all about sample preparation artifacts. There is interesting biology that we can tease out through this multi-experiment comparison of most enriched mass shifts PTMs in a particular experiment. Now, I want to conclude with a few thoughts on another application of this massive set searching that's really terrific for that purpose, which is Glycoproteomics and Ms. Frager Glyco-mode that we published last year in Nature Methods. And again, we take advantage of ultra-fast indexing, and it's fully supported in FragPipe, including false discovery rate estimates that the glyco sort of mass shift specific, which is important to think about false discovery estimates in this context. So we added to this conventional, not conventional, to our massive set open source strategy specific features for glyco analysis, such as support for all activation types, including this, you know, in HCD, we expect to see this sort of the 2 or 3 delta mass shifts on fragments instead of full fragments, full glycans, as well as, you know, checking for diagnostic oxonium ions in the spectra, sequence motifs for handling glycopeptides and so on and so forth. And I want to say that all of this is also applicable to other labile modifications, not just glyco. And in fact, we are working on this very actively. So we can do massive set searches, we can do open searches, and I think it works really well. It's not perfect, but we are working to improve it more. If we apply it to data sets, such as recent data sets, for example, from Nick Riley's lab, Nick Riley is the first author where they looked at Glycoproteomics here, comprehensive and glycoprofiling, and in the original work they used Bionic. So we identify with MS-Fragger massive set search, identify almost 40% more glycoproteins and glycosides compared to Bionic, and it takes us just 1 hour to analyze all the ton of data that Nick generated there. It's minutes per file compared to very, very long searches with other tools. So I think that's really a good advantage as well. And here's some statistics how much we gain as far as the number of glyco-PSMs for HCD spectra as well as for hybrid AI ETD spectra. So we see quite a big boost. Now, it's also applicable to all link glycoproteomics. So here we are illustrating this using the data from this particular publication from Hui Zhang's lab. 24 fractions, different organs, kidney, serum, T cell samples, HCD fragmentation on QE-HF, and compared to the original analysis in this paper with sequest, you get 4 times as many glyco-PSMs, because what we do here, we do an open search, find all sort of detectable glycan compositions, in this case about 300, and then we repeated massive set search with this 300, and we have 4 times as many PSMs, more than double the number of glycopeptides and significant boost in number of glycoproteins. And most new PSMs contain multiple glycans. What's actually interesting that we observe multiple glycans present on the same peptides, because this operator enzyme initially wasn't expected to cleave in general after, it was expected to cleave only after glycoside. So that's certainly not the case based on our analysis. Now, we fully acknowledge the limitations, or rather the challenge is that we are not really doing the, the full job now, characterizing what kind of glycan we have. We're doing a really good job with the peptide sequence and the total glycan mass on the peptide that we can identify. We are working on how to do better as far as figuring out what exact glycan we have, where it's exactly located, as well as the composition of individual glycans, and if you have multiple glycans on the same peptide. So it's a challenging problem in all-link glyco data, but technically, we will get there because we're actually working on that, adding site localization and so on. So to summarize, advantages of FragPipe computational platform in general, I think it's a really a platform that enables so many different things, and I think it's doing each one of them well. It can do comprehensive PTM profiling using open and massive set searching, as I described in my presentation. But it's also really great for applications where searches take too long. For example, peptidome data, HLA peptidome, endogenous peptidome, including searching for post-translational modifications on those peptides. In proteogenomics, we also extended to a timsTOF data, Orbitrap FAMES data. Importantly, we have IonQuant for LFQ with match between rounds and FDR control, as well as SciLac, IonQuant, TMT integrator from TMT. You can build spectral libraries for DIA data, including DIA Empire computational tool as part of FragPipe. So it's a really a complete computational platform that you can download from the website that you see listed here. So I'd like to acknowledge my lab, and especially Finchao Yu and Dan Pulaski, who are working on MS-Fragger and MS-Fragger Glyco, and Danny Geisler on PTM-Shepherd, but my whole lab as well, they're listed here, as well as alumni of my lab, listed on this slide as well. And many collaborators, our lab is really driven by collaborators like Eliane and like others who push us to develop these tools for challenging applications where existing tools may not be adequate. So if you have something in mind, please contact us. We love when people contact us and push us to improve our tools and give us feedback, positive and negative. And finally, acknowledge our sponsors, the NIH and other sponsors. So thank you so much for this presentation. I hope I have a time for a question or two.
Alexey, thank you so much for this wonderful talk. And we have quite a few questions in the Q&A. And I'll just quickly also say to all the attendees that our panelists and speakers have kindly agreed to stay 15 minutes over time just to kind of start to take a few more questions. And we have a few slides with a couple of forward-looking points about PTMs. But let's first kind of go through some questions for Alexey. So from Martin, how do you control FDR in an open search?
So we extended a massive set mode of peptide profit in such a way that basically we, for each mass being of say 1 Dalton or 25 or 26 or 157, whatever it is, we model a distribution of scores. So basically we calculate posterior probability of each identification separately for each being. So if you have a being that corresponds to a very abundant post-translational modification where we have a lot of high scoring PSMs with that particular mass shift, our model would assign high confidence to those peptides compared to a mass being where we don't expect any PTM or it's a very unusual PTM, and we see a lot of sort of low scoring, including decoy matches in that being. So our posterior probability calculation is mass being specific, so that allows us to basically calculate FDR in a PTM specific way, kind of in a mass shift, which is sort of equivalent to PTM specific way.
Another question, a very basic question on open search. Wouldn't it increase the chances for reverse database hits as well? In such case, is there a difference in FDR validation that needs to be employed to distinguish false positives from true positives in such open searches?
Well, again, we demonstrate, I can tell you that while it increases the search space with appropriate FDR control, it does not increase the false discovery rate because we are controlling that through this extended massive set search. So it's a very, very complicated question that I'm not able to address here fully, but I'm happy to discuss with others in offline mode or read our publications. We pay attention to that. It's a very important question.
And some of the questions we'll actually keep for the discussion because they connect to the points, but I'll ask just another one. Is TMT Pro 16-plex phosphor supported in FragPy version 15 or only proteomics at this stage?
No, it is supported, yes.
It's supported. I'm looking if there is just one very quick question that it's specific. Do you think MS-Fragger could be used to identify, characterize new PTM motifs, or for known PTM motifs to integrate information on motifs into the original peptide search, already providing information on which motifs are enriched in a given biological condition?
Right. So it's a good question. The second part, certainly once you identify peptides, you can see what motifs are enriched, right? Now, as far as taking motifs and using them as part of a MS-Fragger, well, for end-link glyco, it's a simplified example of that because we have an option to restrict our glycosylation events only at the sites that have an N and then something SOT motif, right? So we are restricting that to sites that conform to a certain motif. Now, we could extend it even more in a more elaborate way by giving the user ability to specify motif of interest. It's not implemented yet, but if someone has a very interesting biological application, contact us and we can implement that. Like I said, our lab is pretty much collaboration driven. So there is nothing that we cannot implement if there is a good motivation for us to do that.
Alexey, another question. Cross-link cleavages could give linkage information in glycoproteomics. Does MS-Fragger have the capacity to search for these complex, often isobaric glycosidic ring fragments?
Okay. You could specify any mass shift on a peptide. So, there is no, you can put up to 2,000 or 3,000 Daltons if you do fully open search, and you probably will pick them up. If you know exact masses, you can go through massive set search. So we have not found an application yet where we cannot apply open searching, massive set searching as it is, or that we cannot modify our algorithms to do it. So pretty much everything is possible. I think it's such a versatile strategy.
Okay, so we'll keep some of the other questions for the discussion time, just so that we have a chance to get to some discussion. Alexey, thank you again. And we thank all the speakers for these wonderful talks. So you would normally hear lots of rounds of applause from everyone. Okay, so we'll continue now with a few points of discussion. And I will encourage all attendees to attend. If you have something that you really wish to talk about, raise your hand maybe, or put them in Q&A maybe. I'm trying to see how I get to see this here in this format, in this presentation format. So we put together a few bullet points for discussion now that we heard all these great studies on PTMs, a little bit future looking at PTM studies and what are the opportunities and what are the challenges associated with post-translational modification studies. And I think that all of our comments could be synthesized in three different areas. One that deals with the diversity and regulation of PTMs. One, the promise and the clinical opportunities for studying PTMs in health and disease states. And the third one is how can the databases available, how can these resources be improved, and some considerations about data format in proteomics or for PTMs. So we'll start with the first category, PTM diversity and regulation. And one of the question was, how far are we from integral view of the cellular PTM landscape? Can we estimate the total complement of the proteome that includes PTMs? Other points here of consideration are the dynamic, spatial, and temporal regulation of PTMs in a cell. How far are we from understanding these? We would probably need to really need to consider the biological context. Quite a few of the studies that we heard about today, including the one from Laura, where modification was found during infection and then found to be relevant during cell cycle or DNA repair. So there may be many modifications out there that are identified in one given biological context, but they are actually functional and relevant in multiple circumstances. What is the crosstalk between PTMs and the value of open search strategies that you just heard from Alexey? And can we integrate this knowledge of PTMs with protein abundances, protein interactions with other type of information? How far are we from actually doing this in a meaningful way? So these are some points brought up, and I'm gonna also ask a few questions that were not addressed earlier because they connect quite nicely to some of these questions. So one question that came out during Nicki's talk from Michaela Delcha, how does the glycosylation influence the protein dynamics, opening or closing of proteins, activation or inactivation of proteins? Is there any relation, more glycosylation, more stable, more active? And I think that comes especially to this dynamic regulation of PTMs and their functions. Nicki, maybe you can address that question.
Well, I can try. As I said, there's many functions of glycosylation that we don't know about and mean that we do. But I tend to think of it much more in interaction rather than switching it on and off. And what we're increasingly finding is that there are circulating glycosidases in plasma and in ECM, etc., that are just sitting there basically waiting to something to tell them to change the glycocalyx. So this was not previously, these were thought to sit in the Golgi and in the, internally in the cell to turn over glycoproteins in the lysosome, etc. But they're increasingly being found extracellularly, including working on exosomes, which relates to the other one as well. So I'm seeing that the dynamic turnover is actually quite quick because the enzymes are there to modulate the structures on the surface. So we have to wait for them to be added to a protein during synthesis and secretion or movement to the membrane. They actually happen I believe, in real time.
Thank you, Nicki.
I'm not sure if that answers the question, but in terms of stability, I'm not sure that that's ever been tested.
Yeah, and maybe I'll bring that first bullet point and ask you, Alexey, your view, since now you're finding all these new PTMs, and we heard other talks talking about unconventional PTMs. Claire, you can comment on this as well. How far are we from actually getting an integral view of the post-translation modification landscape? Alexey, what are your thoughts?
Yeah, well, I think the challenge is that a lot of things that you find through open search and other modification searches are common sample artifacts. So there is still a challenge of getting to truly interesting biological modifications because they're often in the grass. So plus what we also need to do is to make these search strategies more quantitative so we can quantify unexpected modifications through across different samples, which is challenging, you know, because the conventional search strategies for, like, say, label frequency is match between runs. You know, they haven't been explored yet for this open extended modification search strategy. So I think we're getting there, but there is still quite a bit of computational work that we need to do to really identify and quantify low abundant interesting modifications. So now we are doing a good job at finding sort of more abundant modifications that we not necessarily expect in the samples. Finding low abundant unexpected modifications, I think, is the next next frontier.
And there is a question in the chat from Martin that kind of gets to this. The general question: site localization software in many database search software programs increased the list of phosphopeptides significantly by localizing several sites per peptide. In most cases, quantification of these isoforms are exactly the same in large-scale quantitative studies, indicating artifacts. In addition, this random site phosphorylation strongly contradict kinase motifs normally seen in kinase substrate interactions. So do you think that these programs should be used, or can we improve them to get a more accurate picture of the phosphoproteome? And maybe Andy can comment on this as well, or Claire, I see your hand up. So Claire, maybe you can go for this.
Yeah, if I understood the question correctly, I would query how they're using that information, because site localization algorithms will give you an indication of ambiguity. And if it's suggesting ambiguity, then that is about identification, not necessarily which site is present from the biological material. Can I go back to the discussion about the previous question that Alexey was talking to in terms of how close we are potentially to identifying the PTM landscape? I think we're still, you know, it's the tip of the iceberg. Mm-hmm. And I think some of the studies that we've done on atypical phosphorylation and are still coming through looking at different sites of modification, sulfation we're now working on as well. From an analytical perspective, and this doesn't matter if we're talking about mass spectrometry or any mode of analysis that we do, we are so constrained by how we prepare samples and how we use that analytical instrumentation. And unless we cover all eventualities in terms of, for example, biological sample stability, dynamics, you know, have you chosen exactly the right time point at which to harvest that sample, you know, diurnal cycles, whatever it may be, we're not going to identify them all. So I think we need to be cognizant of that fact. It's a lot about sample preparation and sample acquisition. With regard to Alexey's point about, you know, finding the needle in the haystack and the fact that what we're seeing are the most abundant, something that I wonder about a lot is actually whether or not we do proteomics arguably in completely the wrong way. We are very different as a community to people who do metabolomics. So metabolomics, they work out what changes and then they try and figure out what it is. We try and identify everything and then figure out if it changes and therefore is interesting. I think if we as a community moved more towards what metabolomics do and say, actually, we don't care what it is unless we know that it's changing in response to, you know, condition X, Y, or Z, disease, whatever, we actually might make a lot better use of our available resources. Anyway, stop.
So one other comment that I would add to what Alexey and what you said, Claire, about maybe potential random artifacts and what is biologically meaningful. And I feel like we in this community have had this discussion for quite a while, right? Okay, you identify a modification, but is it, what does it mean? And does it have any function? And sometimes, okay, we make some standard mutations and maybe you find, maybe you do not find a function. And I'm wondering how blind some of those approaches may be if we consider that maybe a modification would require the coexistence of another modification on that protein, and how far we are from actually capturing truly the state of a protein and the multitude of modifications that can coexist. And I feel kind of coming back to Claire's point that many of the approaches that we use are actually blindfolding us a little bit. Let's say we do a phosphopeptide enrichment, we do an acetylation enrichment, we do whatever our favorite or whatever our question of interest is at the time, but they are fairly challenging. So they sort of open search approaches that Alexey was talking about could partly tackle that. Claire, I see you unmuted yourself, so maybe you would want to comment.
Yeah, just to comment on the fact that I think open PTM searches are probably the way to go, because we constrain ourselves so much by saying, you know, am I just going to look for phosphorylation, or am I going to look for sulfation as well? And actually, depending on the tolerance you use for that, you may identify a whole load of things incorrectly. But you know, you're going into the realms of doing top down now to look at combinatorial, and you know, we have been talking about this for more than a decade, and you know, we haven't got there. It's really, really difficult, and there are so many I guess snag points on that pipeline, and we've still got so much basic work to do before that is in any way routine, I think. So I think for now, individual open PTMs might be a better way forward for most of the community, at least.
And I have to say, we had also a question about top-down approaches in the chat. So one thing that we will try to do is maybe set up a Slack channel with all the questions that we have had, and maybe a line of communication between all this group. And I copied all of your questions, so nothing was lost, and we'll do our best to try to create an active interaction and discussion even after this webinar and between webinars. So I'm just going to go to the next slide in the interest of time, that kind of now transitions to a clinical and health related perspective on PTMs. So here are some of the questions. Translation of complex PTM pattern dynamics into phenotypes in health and disease, which obviously it's really critical and provides quite a bit of promise, and we touched on this a little bit in some of the talks. So maybe I'll ask Boris to comment on some of these aspects.
Yeah, thank you. So, well, what here occurs to me, and it was mentioned already at the very beginning, the proteogenomics, right, in terms of cancer and so on. We have now a kind of boom of studies, also large-scale studies, and especially in terms of cancer, understanding how individual mutations actually change the PTM landscape, right? So that is something that is very interesting and super difficult to analyze, obviously, by mass spec, you know, to hit all these mutations, but bioinformatically, it can be addressed. So obviously somatic mutations changing the landscape, it can be in a very individual manner, and it can, you know, point to, you know, increased activity or so, or decreased in specific kinases, for example, right? So this is what occurs to me as maybe not a direct application, but like a link to individualized proteomics, right? In terms of the those. And I mean, in that context, of course, targeted proteomics, in case you really want to detect those, right? Again, very challenging still, despite of PRMs and you know, all sorts of things, at least in our hands, proves to be very even time consuming. But I have to say, we are not really specialists in targeted proteomics. We did try to do it a couple of times, but then kind of bumped into quite laborious workflows and so on, right?
I can add to that. So we have in my lab put quite a bit of effort into developing PRM assays for detecting different post-translation modifications and kind of monitoring them during infection, which, yes, as you said, Boris, time-consuming, a lot of experimental validation needed, but in the same time, way better than having to develop all those antibodies. So there are pros and cons, right? And then afterwards, still we do all the studies in cells, and then we come back to other questions and other challenges for how to monitor these. But as you said, it's challenging. And Laura showed one example of a PRM on an acetylation, but what she didn't quite confess that is that she needed to do an acetylome enrichment just to get to that to do the PRM on that. So it was quite an expensive experiment for one quant. So how, Andy, maybe I'll ask you to pitch into those last two bullet points on this slide. PTMs to drug, need for a proof of principle beyond kinases, which PTM for what disease, and you're okay with sticking to kinases if you want to.
Yes, so we do have actual project going that direction. Is I certainly feel that especially tyrosine phosphorylation and a lot of these targeted, certainly it is a kinase inhibitor. I think this in terms of phosphorylation is that tyrosine basically kinase inhibitor is still a major directions, and by companion, for example, by looking at some of these targets and can quickly check the status of the phosphorylation to evaluate the clinical outcome and also I think beyond the therapeutics and for long-term monitoring. I think these are certainly have the application. I do see from my lab point of view that great applications, but certainly a lot of challenges. For example, I mean, beyond a lot of people talk about these PTMs that unfortunately beyond mass spec field, all of them are still considered antibody as a way to go. So make it so hard for us to follow validation, and then a lot of PTM antibody are not available. So that's put another layer of difficulties. And then on the top of that is certainly Alexey now talk about low abundant. Now I'm just have all the headache even for high abundant PTMs. So in terms of validation is so much harder than the regular proteomes. So there certainly is a lot of challenges. For example, we do these days do a lot of validation for clinical samples, but we need a lot more samples compared to the standard, more like a whole proteome. So indeed, it's a lot of challenges if we focus on the PTMs and especially if we want to get based on site specific. I believe certainly there are a lot of certainly, I would say a great opportunity, but at the same time it's a lot of challenges ahead, especially moving into the clinical field.
So Ferdinando, maybe very briefly you could add on this, because you started us off this morning or evening for others, very late night evening, with some consideration about clinical applications.
Yeah, I mean, I think well, first of all, we had so many input in the meeting, in the talks we heard today, that I mean, this discussion could and should be going for hours. But I think the link for the medical or clinical application is maybe a bit going in the direction that Claire pointed out, but also Boris and Andy. So how can we approach an information-oriented way of seeing the biology? So what are these PTM doing? What is the goal into something which can be helpful in treating the patient, and which drugs? So going for the broad question to maybe trying to see which specific question could be addressed by applying the technology we have heard today. So as an example, if you have a specific drug which is focused on a certain genetic modification, can we use this information to address why is it not working in certain condition or after certain condition? and why it's not working still when we know that the mutation is present. And I don't know if it's specifically targeting PTM could be an answer to address. And obviously phosphorylation is definitely more interesting because so many drugs are focused on that. But Nicki showed us the example of Tamiflu and other viral-directed drugs, which are totally away from phosphorylation, which are really important too. So maybe there are also other sources that we could try to address as drug targets. Yeah.
Thank you, Ferdinando. And just in the interest of time, let's move to the third part. Lots of comments that we received also from the panelists here prior to the webinar were about PTM resources, these databases, and also data format. Like the specialized PTM databases, how complete are these, how representative of the numerous types of PTMs, and how to collect all this information about modifications with the relevant disease metadata. So maybe someone can start to tackle what sort of information is included. Who would like to have a go at this? What is missing? What should be included in these databases? Go, Nicki.
Yeah, I think that what I'm discovering is that we really have to go with databases that are already existent and expanding them. Getting funding for information storage and data storage is very difficult. And there has been so many cases where databases have been made and then they disappear because they're not maintained and they're not. So I think we need to be able to talk to the databases that have already started and get them to expand their content and then link them together so that they talk to each other, ideally.
Yes. Alexey, what's your thought on this?
Yeah, I agree because, yeah, database maintenance, it's extremely hard to get funding for maintaining anything. If you look at the there are so many databases developed for phosphopeptides, for example, and I bet that pretty much all of them are not updated. Phosphosite Plus, which is a sort of commercial support in some way, I think is the only one that's useful for that. So I agree. I think working with existing databases, SwissProt for sure, NextProt as well, that's the way to go. But I don't know, because annotating known modifications, at least we have some idea how to do some kind of quality control. But as you go again to this last explored modifications, how do you control the false discovery rates? Because if you submit data sets and there is no good way to score those submissions to those databases, they'll be flooded with useless data. So you have to also develop metrics that we need to apply for identifications that are coming into those databases, should that happen.
And there's a comment in the chat, and please don't fill up the databases with poorly validated data on PTMs, which is a really valid point. Absolutely. Frank, I'm gonna put you now on the spot because the next slide is something that Frank proposed to discuss also. How many file formats and programs do we need?
Yeah, it was just, I came up with the point, so when I'm teaching my students, so how can I identify PTMs? So, and then if you just look in the literature in the past years, it's amazing how many new tools we have, how many new formats we have. And then I guess a couple of weeks ago, we got a new spectral library format, the 7th ones. So I'd just like to ask you, so how we can make it a little bit better. I mean, so I just took this simple example here from Edward Tufte. He said, if we have more than seven different colors, for example, then our brain is overloaded. And we have now so many different tools. And today, even also we saw FragPipe, we saw MS Fragger Glyco, we PTM-Shepherd. That's we need. I guess the community, if you are starting doing PTMs, we need a kind of orientation and maybe a unique file format. Like with it was a big advantage with the PEP XML, for example. So we could use this file format in each program. So what are your guesses? So
and we know that the genomics, the field of genomics has been through a similar debate earlier on, but of course with proteomics we have this challenge of increased complexity, increased data types, proteomic data types, and yes, so.
But maybe I like to ask Nicki. Nicki, you have a paper in the pipeline which somehow compares different programs, and so what was your experience?
That's an interesting question. Let me put it this way: half of the participants used the same software and got a range of different answers from the same data, which is saying that the search parameters that you use, the restrictions and the parameters that you do to restrict your searching so that you don't get lots of false discovery plus inaccuracy are really essential. So I think I agree with you. What is needed is almost a primer in how do you do this with this software to make it correct. And from my experience, proteomics went through the same thing, that at the beginning of proteomics, they were identifying, lots and lots and lots and lots of proteins, which turned out not to be true. And then false discovery rates were brought in, etc. So I think we're doing the same thing now. We're sort of finding all this stuff, which when we go back and look at it, may not be correct. So it's a process, I think, and we should learn from what's gone before, I think.
Alexey, what are your thoughts?
Well, first I would say that we do need multiple tools because we validate our own tools by looking at how other tools perform. So even though we are, you can say, competing with other labs that develop computational tools, we're also grateful for their work, because without having that, we wouldn't know how well we're doing. And so we need that redundancy to sort of innovate, and that would push us. And another comment is that, for some applications, I think commercial tools were dominating the field, and they're extremely expensive. So we do have to have academic tools available. And again, in the nature of academic funding, that we have to have redundancy, because there is no guarantee that any of us will be able to develop these tools, forever, because it all depends on funding. So we have to build in some redundancy into that. So yes, biologists have to suffer through the fact that there are many choices available, but hopefully the field will continue converging on things that work. And again, it does not have to be one tool. It's more important that we have guidelines for how to best use each tool that's available.
Yeah, and yes, and I think that the guidelines have been so important in this field for promoting the proteomics field and gaining credibility for the proteomics field in different biological and clinical fields of study. And it has been great to see MCP and JPR and all these journals that have really asked for guidelines and implemented guidelines, and also the HPP that has been so important in setting up guidelines. So on that note, I'm going to go to this slide and pass the buck to Chris overall, who has been waiting to tell us a little bit about a special issue. So this is really an important annual event for the Human Proteome Project, this annual special issues in JPR. And last year, many of you may be aware of the fact that there was this milestone reached with about 90% of the human proteome reported, and this was celebrated in the Nature Communication paper, and a special issue, virtual special issue put together, and another JPR issue. And now it's going to be the time for a special issue this year. Chris, you can share.
Yep. So just briefly, the JPR special issue has been going for 9 years now, and it's been a great vehicle for publicizing progress on the Human Proteome Project. It started off, as many of you know, focused mainly on the CHPP, but we have projects and papers in progress, and we've expanded that, of course, to the BD now. And what was nice, as Ileana just mentioned, last year, it was reported that we had 90% of the human proteome discovered at PE1 existence, and now with the latest release in January this year from NextProt, we're down to 7% only is missing. So we've got 18,357 proteins at PE1 level. So we've had several, this is the cover of last year's one, special issue. The point is that there's papers in this issue receive a huge number of citations. So it's one of the largest, it turns out for JPR, one of their big boosts in impact factor. So your papers actually get a lot of coverage here. And just finally, the deadline this year, because of the pandemic, is later. It's in June 30th. So please, we're trying to encourage the D-HPP to submit papers that can be related to progress on the human proteome. But in addition, we started some years ago trying to get the various BD teams to look for emergent proteins that would be in response to a disease or to cytokine stimulation or healing or in remission phases or in development that would only appear in very, very limited windows. And these types of tissues are not normally captured by general proteomics people. We just encourage you to look for missing proteins in those data sets. And you don't have to write a full paper or take your Cell or Nature paper and put it into JPR. But we've got a format for just a single one-pager, a letter format, where if you found good evidence for a missing protein in cancer or in microvesicles or in some strange or rarely studied tissue or developmental process, please have a look there for missing proteins. And these are easily accessible in lists on NextProt, and you can quickly pass through your protein lists and quickly gain for some postdoc or student in the lab a one-pager or two-pager, and that's a good CV booster, and also it's just nice for the community. So we look forward to your papers, and we thank always the help of the guest editors, because in addition to the normal review process, which is 2 to 3 externals, we also get your papers are also reviewed by several of the guest editors. And as many of you know, Gil Oman is one of the toughest of the lot, and so your papers pass through that. So it's not an easy route into JPR, but it's these are very nice papers. So thanks very much, Ileana.
Thank you very much, Chris. Okay, and if you can stop sharing the screen.
Mm-hmm.
I just want to thank again all the speakers, and I want to thank our audience for staying this extra time, and please send us your suggestions for themes for future webinars, potential speakers. We want to hear from you. Format, obviously we want to change this to make it fit everyone. So just send us your thoughts, and we'll try to also implement a Slack channel, or we'll brainstorm a little bit what would be the best way to keep interacting and answer some of these questions, even in between webinars. And again, thank you very much to all. And I know it started very early for some, and it finishes extremely late for some. I think for Nicki it's 3 a.m. in the morning now. So thank you again very, very much. Okay, bye everyone.